something. Hey, that is let's look at that. One, and two, and three. At least 10 inheritance comes over inheritance. Yeah. That is tomorrow and evolution and evolution matters. Oh, that's an 11. No, it's a 10. That's a 12. It's also power energy electrons. This is a 13. Sure. This will be for 3 lessons.
there. The first step is to go from the gene to messenger RNA. That the RNA, the messenger RNA, you can view it as a transcript. We have rewritten the information now as RNA. And then the next step, which we're going to dive into in this video, is going from that messenger RNA to protein. And this process is called translation, because we're literally translating that information into a polypeptide.
Organic. Positive of the gene. Transfers. Phosphate groups. Transfers. Molecules. Kinase. Km is the substrate concentration. This is proline, pro, P, hydrophobic, polar, breaks up, breaks, is kinky, breaks up helixes, found in beta terms, disrupts helixes, amino acid. It will link molecules together using ATP, fourth bond using ATP, fourth bond using ATP. It will attach the nucleotide. It will attach nucleotide to the sugar phosphate backbone, right? Essentially, the nitrogenous base will make sure it links to the sugar phosphate backbone phosphodiester bond. Tyrosine TYR. Tyrosine TYR. Y. Cytosine restricted slightly polar, aromatic. Is used to attack the phosphate groups. Vmax is the maximum speed at which the maximum speed in 1 second at which the enzyme will turn substrate into product. Maximum rate. Allosteric effector binds at the allosteric site and induces exchange. Allosteric inhibition effector. Allosteric inhibition. negative effector. Arginine with a guanidinium group, arginine, ARG, are very basic, very polar, such an intersect guanidinium group. This is the Michaelis Mendelus. Wait, yeah, Michaelis Mendelus. Michaelis Menten. VF stands for the maximum velocity of reaction. Needs to be greater than the enzyme concentration. This is glutamate, GLUE, scientific characteristic, negative. Negative. Glutamate longer than aspartate by one carbon. Acidic. Polar. Cofactor. No. Coenzyme. Organomolecule. It will transfer electrons. It will carry electrons in transfer functional groups. This is the guanidinium group found in arginine. Cooperative enzymes, cooperative binding enzymes, cooperative enzymes. When there's a neutral side chain. Neutral side chain is pKa1 plus pKa2, where pKa1 is the carboxyl group, pKa2 is the amino group. A kinase transfers phosphate groups is in the class of transferases. Adds the phosphate group from A to B. ADP plus B. Cooperative binding is when sigmoidal, it causes the it can cause the enzyme affinity of the substrate to go up or to go down, more or less likely to bind. Yeah. Phosphatase removes phosphate groups in the class of hydrolase. Catalyzes enzymatic reactions that involve redox reactions. That involves redox. Oil rig. Also transfer electrons. I think that's good. So enzymes do not alter the delta G or delta H and the final composition. The active site is a site of binding from the enzyme to the substrate, cytokatalysis, is written N terminus to C terminus. So, yeah. This amino acid is so amide, and then we have glutamine. This is glutamine because the extra carbon. Glutamine, GLN, GLN. Sorry, glutamine, GLN. Oh, are you serious? Glutamine, GLN. What is your name? What is your name? Q. Your name is Q. Cytosine characteristics. Polar. 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 Hydrophilic. The cofactor is an inorganic. Oh, it's an inorganic cation that will help enzyme function. Metal cation, metal cation. L and D indicate relative configuration. S and R indicate absolute. L and D thinking like family traits or something, and this one think of the exact address of the atom, the exact address of the atom's molecule. Amino acids are some organic compounds contained both in amino group and a carboxyl group and an R group. Proteins convert motor proteins, such as muscle contraction, physical movement, and cell motility. Motor protein, cargo. Is an organic small molecule that will carry electrons or transfer functions. Isoleucine, Li, non-polyphatic. So isoleucine, we have amino acid, carboxylic group, amino acid, carboxylic group, hydrogen, and then we have this is butyl, so we're going to branch early at the second carbon. So carbon, branch, CH3, and then H, and then and then carbon. So make like a little check mark. Yeah. Okay. Hydrophobic. Kalium is the substrate concentration. Hydrolase that are not unaffected by the unaffected, unchanged in uncompetitive, Uno competitive, game goes down, uncompetitive. We also have parallel lines in the Limburger pot, and yeah. Uncompetitive binds to ES only, at the active site. Non-competitive is ES or E and parallel. PM is the maximum reaction rate of the enzyme at given us in 1 second. Is when there's a basic side chain is pKa 2 plus pKa 3 over 2. Uncompetitive, Vx max goes down, Km goes down, parallel. Binds at the allosteric site and binds and lowers binding affinity. Induce a change in the conformation of the enzyme, so that it can no longer bind to the active site. Michaelis-Menten equation. VO is the speed, the given speed, the current given speed. Vmax is the maximum enzyme to substrate catalysis. Essential concentration plus Kn, essential concentration given half of the Vmax. It also tells you the binding affinity of the enzyme. We have to make sure that substrate is greater than the enzyme concentration, and binding affinity does not depend on enzyme concentration. Okay. This is methionine. Meth, M, polar, hydrophobic, contains the thiol group. An allosteric effector binds at the allosteric site and does not allow the substrate to bind to the enzyme due to conformational change in the enzyme, and lowers binding affinity. This is histidine, his, H, polar basic hydrophilic, contains the imidazole. Histine is neutral at physiological pH, pKa 6. A cofactor. Zymogens are what are zymogens? They're like pre-enzymes, I guess, like pre-enzymes in a state of pre-enzyme. Oh yeah, precursors to enzyme. Okay. Cooperative enzymes. Cooperative binding is when the enzyme's affinity to bind the substrate goes up or down, which is dependent on yeah. With water, catalyzes cleavages with water. pKa plus 1 plus pKa plus 2 divided by 2. Water proteins convert ATP into mechanical movement by the hydrolysis of ATP. Chemical energy into mechanical work. Chemical energy into mechanical work. Chemical energy to mechanical work. Really interesting. Precursors to enzymes. Aspartate, ASP, ASPT, negative polar hydrophilic, acidic. Reaction scheme is VO is equal to Vmax plus substrate concentration, time substrate concentration divided by Kn plus substrate concentration. Oh, okay, so reaction scheme. Yeah, okay. This is glycine, G, nonpolar, hydrophobic, is a chiral, rotates, disrupts alpha helices. Yeah. Motor breaths will convert chemical energy into mechanical work by the hydrolysis of ATP, means it uses ATP. Oh no, hydrolysis, never mind. Hydrolysis by use, yeah. Muscle contraction, testicle movement simultaneously. The exercise is a site of catalysis. But it gives you where the reaction rate is half the Vmax. And it also tells you like about binding affinity. Catalyzes substrates by changing by interconversion of isomers. This is the imidazole group. You see the reaction rate of the enzyme, the maximum reaction rate of the enzyme occurs in a second. Catalyzed enzyme, yeah. Turn substrate into products. Oh, it's also when the enzyme acts are saturated with substrate. That's true. Middle category. Inorganic. Valine is amino group, carboxyl group, and it'll C and then it'll fork. Carbon and then it'll fork. Hydrophobic, yeah. e, Imogen, 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 Zymogen. Without water or redox, we have um, we have aldolase and carboxylase, aldolase and decarboxylase. And we can go again. That is organic. It carries electrons or other functional groups. Hydrolase, uncompetitive, is also parallel on the Lineberger plot. By hydrolysis, by hydrolyzing ATP, hydrolysis of ATP, muscle contraction and circumference cell motility. Catalysis is cleavages, also forms all bonds and cyclic structures, decarboxylase and aldolase. Exergonic at the active site. No, binds at the allosteric site and for an allosteric inhibitor, it's going to change the conformation of the enzyme and make the substrate not able to bind to the enzyme. Activity goes down. Our precursors to enzymes is E plus S turns into ES plus K1 plus ES plus P, and that's K2, and then ES can to ES plus 1 by K minus 1. Two is the rate limiting step. Chemical energy to mechanical energy. It gives you, I told you the binding affinity of the enzyme. It also is where the substrate concentration is at half of the Vmax. And the reaction rate is half the Vmax. Zymogen without water or transferred electrons, decarboxylase and aldolase. Generates double bonds and restrictions. Maximum rate at which the enzyme is turning substrates into products. It's also where the enzyme is most saturated with the substrate. All enzymes are saturated by hydrolysis of ATP, such as muscle contraction, vesicle movement, cell motility. Okay. Do President have anything to do today for IU? monkey's done. Let's now go ahead and do eggplant.
Sequence. And you can see a little.
Let's turn the light a little bit.
Little bit visually here. This is all review. We covered a lot of this in the video on transcription, and the overview video on transcription and translation is if you look at a eukaryotic cell and the bacteria in a prokaryotic cell, it's analogous. You just don't have the nuclear membrane, and you're not going to do the processing step that I'm going to talk about in a little bit, and we went in detail on the video on transcription. But you start with the DNA. You have your RNA polymerase as the main actor that's able to transcribe the RNA from that. If we're talking about a eukaryotic cell, what you end up with, we wouldn't call mRNA, we would call that its pre-mRNA. Pre-mRNA, which then needs to be processed. The introns need to be taken out. We add a cap and a tail here. And if we're talking about a eukaryotic cell, we'll then formally call that mRNA. And then it can travel, and this is where we get into the translation step, it can travel to a ribosome, which is where it will be translated into a polypeptide sequence. And you see the analogous thing happening here in this bacterial or this a prokaryotic cell right over here, except you don't see the nuclear membrane because this is prokaryotic, and you don't see that processing step. So you could just consider this straight. This is mRNA right over there. So the questions are, well, how does this thing happen? And what even is a ribosome? So let's zoom in a little bit on a ribosome right over here. And there's a couple of interesting actors. One, as you can imagine, is the ribosome itself. And it is made up of proteins plus ribosomal RNA. So in the video on transcription, we're already familiar with messenger RNA, and we often view RNA like DNA as primarily encoding information. It's acting as a transcript for a gene, but it doesn't have to only encode information. It can also, so it's proteins plus, that's not a T there, this is a plus. It can also provide a functional structural role, which it does in ribosomal RNA. And this big, this looks like an oversized hamburger bun or something right over here. This is a super oversimplification of what a ribosome looks like. And I encourage you to do a web search for image searches for ribosomes, and then you can get a more appreciation of how beautiful these structures are and how intricate they actually are. So this is the site, and you can broadly think of the ribosome as having this, this is the top bun and the bottom bun, and it's going to travel along the mRNA from the 5' end to the 3' end, reading it and taking that information and turning it into a sequence of amino acids. So how does that actually happen? Well, each of these every three nucleotides there, you call that a codon. So that's a codon. This is in a color that is visible on both white and black. So this next 3 nucleotides is a codon. This is a codon. This is a codon. And what's actually, the information is actually encoded in the nitrogenous bases. So this first codon right over here, we see it's AUG. So the nitrogenous bases are adenine, uracil, and guanine. And this has this codon, it codes for the amino acid methionine. But this is a good one to know. AUG, let me write it over here. AUG is known as the start codon. Start codon. This is where the ribosome will initially attach to start translating that messenger RNA. And so the way that this drawing is that we are just starting to translate this messenger RNA. So how does that actually happen? How do we get from these 3-letter sequences to specific amino acids? Well, let's think about it. How many possible 3-letter sequences are there? Well, there are 4 possible nitrogenous bases there. So there's 4 possible. So if you have a codon and it has three places, there's 4 possible things that could be in the first place. there's 4 possible things that could be in the second place, and there's 4 possible things that could be in the third place. So there are 64 possible permutations. 4 times 4 times 4 permutations. So you can think of it, there's 64 different codons, different ways of arranging the A, the U, and the G. And that's good because there are many amino acids. And this is actually overkill because there's actually 22 standard amino acids, 22 standard amino acids, and 21 that are typically found in eukaryotic cells. So we have more than enough permutations to cover the different amino acids. And it's not hard to find tables that will actually show us what the different sequences, what they actually code for. So you can see here, you can take the first letter, the second letter, and then the third letter. Look at the different sequences, and you can say, okay, look at that. AUG, adenine, uracil, guanine, that codes for methionine, right over here. You could do that with any of them. You could say cytosine, uracil, that codes for leucine. And you can see that it's not just one amino acid per codon, that here you have 4 codons, all code for leucine. And so it turns out that 61 of the codons, let me write this down. So 61 of the codons, of the possible 64, code for amino acids, and 3 play a role that essentially tells the ribosome to stop. 3 codons are stop codons, and you can see them right over here. UAA, UAG, UGA. That's how the ribosome knows to stop translating. So AUG, that's a start codon, and it codes for methionine. So that lets you know that, well, these polypeptide chains are going to start with methionine, and then these characters tell it where to stop. But how does the amino acid actually how do they all get tied up together to form this polypeptide? And how do they get matched up? How do they actually get matched up with the appropriate codon? And that's where we have another RNA-based actor, and this is tRNA. So tRNA, the T stands for transfer RNA. There's a bunch of different tRNAs that each can bind to specific amino acids. And on parts of those tRNA, they have what are called anticodons that pair with the appropriate codon. So this tRNA, that's not what it looks like. I'll show you in a second what it looks like. That's a tRNA molecule, tRNA. At one end of the molecule, it's binding to the appropriate amino acid, methionine, right over here. And then at the other end of the molecule, although it's in the middle of the tRNA actual chain, you have your anticodon. And your anticodon matches up to the appropriate codon. And so this is how they bump into each other the right way, and the ribosome is going to facilitate it, that the AUG is going to be associated with the methionine. And if we look at what tRNA actually looks like, and this is still just a visualization. So this is a strand of tRNA. You get a sense of, okay, it's a sequence of RNA right over here. This is I guess you say, think that it's 2-dimensional structure, but then it wraps around itself to form this fairly complex molecule. And the anticodon, which is right here, it's kind of in the middle of the sequence, it forms the basis for this end of the molecule. That's the part that's going to pair with the codon on the mRNA. And then at the other end of the molecule, is where you actually bind to the appropriate amino acid. So I know what you're thinking. All right, I see that the ribosome, it knows where to start. It starts at the start codon. I see how the appropriate tRNA can bring the appropriate amino acid, but how does the chain actually form? And you can view this in 3 steps, and associated with those 3 steps are 3 sites on the ribosome. And the 3 sites, we call this the A site. Maybe we'll see if I write it in black. A or yellow. All right, let me write it in blue. So that is the A site. This is the P site. And this is the E site. And I'll talk in a second why we call it A, P, and E. So the A site is where the appropriate tRNA initially bounds, the tRNA that's bound to an amino acid. And so you can see we're starting the translation process. The next thing that's going to happen is another tRNA, the one that has an anticodon that matches the UAU, that's going to bond over here on the A site. And it's bringing the appropriate amino acid with it. It's bringing the tyrosine with it. So why is that called the A site? Well, A stands for amino acyl. You can do, or an easy way to remember it, is the tRNA, it's the place where the tRNA that's bound to an amino acid, just one amino acid, is going to bind on the ribosome. And so once that happens, once this character comes here, let me draw that. Once this character comes right over here, it's going to be AUA, and it's bound to the tyrosine. Well, then you can have a peptide bond form between the 2 amino acids, and the ribosome itself can move to the right. So this tRNA will then be in the E site. This tRNA will then be in the P site, and then the A site will be open for another amino acid carrying tRNA. So what does P and E site? So what do the P and E site stand for? Well, you can see a little bit more clearly right over here. So the P site is where you have the polypeptide chain actually forming. A little bit more clear. So what does P and E site? So what do the P and E site stand for? Well, you can see a little bit more clearly right over here. So the P site is where you have the polypeptide chain actually forming. And so the P site is often, well, one way to remember it is that's where you have the polypeptide chain, and now you have a new A site where you can bring in a new amino acid, and then the ribosome is going to shift. Is once this is bound, the ribosome, the peptide bond forms, and then you have a new A site where you can bring in a new amino acid, and then the ribosome is going to shift. Is once this is bound, the ribosome amino acid, and then the ribosome is going to shift. Is once this is bound, the ribosome, the peptide bond form.
It shifts to the right.
And now P site. So the P site.
So this goes here. This one goes left. Goes here. This one goes left. Okay. This one goes here. This one goes left.
Where you have the polypeptide chain actually forming. And so the P site is often, well, one way to remember it is that's where you have the polypeptide chain, and now you have a new A site where you can bring in a new amino acid, and then the ribosome is going to shift. Is once this is bound, the ribosome,
and then this A will go left and remove itself.
The peptide bond forms, and then the ribosome can shift to the right. When the ribosome shifts to the right,
what's he to the right? Isn't it left?
we're going to be in this position where the thing that was here, that was in the A site, now the polypeptide is attached to it, can shift the ribosome, the peptide bond forms, and then the ribosome can shift to the right. When the ribosome shifts to the right, we're going to be in this position where the thing that was here, that was in the A site, now the polypeptide is attached to it. It is going to ribosome amino acid. And then the ribosome is going to shift. Is once this is bound, the ribosome, the peptide bond forms, is going to shift. Is once this is bound, the ribosome, the peptide bond forms, and then the ribosome can shift to the right. When
this one goes to the right. This goes away, and then this one will what? Oh, and then to this one. Just to the right.
And the ribosome is going to shift. Is once the home can shift to the right. When the ribosome shifts to the right, we're going to be in this position where the thing that was here, that was in the A site, now the polypeptide is attached to it. It is going to now be in the P site, and the thing that was in the P site is now going to be in the E site. It is now ready to exit, and that's why it's called the E site, because that's the site from which you exit. And so this is going to keep happening until we get to one of the stop codons. And when you get to one of the stop codons, then the appropriate polypeptide is going to be released, and we will have created this thing that could either be a protein or part of a protein. So this is very exciting because this is happening in your cells as we speak. This is, and in fact, if you think about things like antibiotics, the way that they work the way that antibiotics work is that ribosomes in prokaryotes are different enough than ribosomes in plants and animals or in eukaryotes that we can find molecules that hurt the function of ribosomes in prokaryotes, but don't do it to eukaryotes. And so if you have bacteria in your bloodstream, and if you take the appropriate antibiotic, it could disrupt this translation process in the bacteria, but not in your cells that you want to keep. So we already know that.
Yeah. Man, what did I just erase?
Let's talk about some of the differences between how translation happens in prokaryotic cells and how it happens in eukaryotic cells. And I want to focus mainly on the mRNA just before it's ready to be translated. So let's start with our prokaryotic mRNA, and let's look at our 5' side first. So we have this yellow part right here, and that's the non-coding region. And it's called the non-coding region because the ribosome is not actually going to read that part. So that particular sequence of amino acids is not that important. And then after the non-coding region, we have the Shine-Delgarno sequence. And bind to. So let's just do a ribosome right over here. This is where the prokaryotic ribosome is going to bind. And then after the Shine-Delgarno sequence, we have another non-coding region. Just going to abbreviate it NCR. And then we have our start codon, which is typically AUG. So that tells us to start. And so the ribosome is going to start translating. It's going to read this entire section, put together the corresponding polypeptide chain until it hits the stop codon, which tells it to stop translating. And then we have another non-coding region. Let's look at our eukaryotic mRNA. And so it's pretty similar, but you can see there are some differences. So we'll start with our 5' side first. So you see this red nucleotide right over here. That's the 5' cap. And the 5' cap is simply a guanine nucleotide. So I'm going to draw a G inside, guanine. And it's going to have a methyl group somewhere on the molecule. So I'm going to draw a methyl group. And the bond between this guanine and the nucleotide right near it is a bond that's different than the bond you typically find between two nucleotides. And so that's really all the 5' cap is. And the 5' cap is actually the ribosomal binding site in eukaryotes. So that means that in eukaryotes, the ribosome is going to recognize this particular part and bind to it. So after the 5' cap, we have this other non-coding region, which the ribosome is not going to translate. And then the ribosome is going to hit the start codon again, AUG. Tells it to start, and it's going to start translating. So it's going to translate this entire section until it hits the stop codon. And then we have another non-coding region. And then we hit something that looks different than what we've seen in the prokaryotic mRNA. So this section with blue nucleotides, and that's called the poly-A tail. And the poly-A tail is a bunch of nucleotides that are all A's or adenines. So I'm going to draw A's inside all of these nucleotides. And the poly-A tail is actually pretty long. So it's typically anywhere between 100 and 250 nucleotides long. So that's pretty long. So I didn't exactly draw it to scale. And the purpose of both the 5' cap and the poly-A tail is to prevent this mRNA from being degraded by enzymes. So it acts as this kind of signal that does not allow enzymes to break it down or degrade it. And so you might be wondering, well, what about prokaryotic mRNA? How come they don't have anything similar to prevent it from being degraded? And the brief answer to that question is that in prokaryotic cells, transcription, that's an R, and translation both happen in the same place. So prokaryotic cells don't exactly have a nucleus. They have this, like cytosol, and transcription and translation are happening in the same place. And not only are they happening in the same place, but they can actually be happening at the same time. So you can have a piece of mRNA that's being formed, and while it's being formed, a ribosome will attach to it and begin to translate it. But in eukaryotic cells, things are a little bit different. So transcription happens in the nucleus, and translation happens in the cytoplasm, where there are ribosomes. And so the mRNA, after it's made, has to travel from the nucleus to the cytoplasm to where the ribosomes are. And so because it's traveling this relatively large distance, it's going to encounter a lot of different things, including enzymes that might break it down. And so it needs this extra protection to prevent it from being damaged in any way. There's one more difference I want to talk about in how translation happens in prokaryotes and eukaryotes, and that is what the first amino acid in the polypeptide chain will be. So in prokaryotic cells, the first amino acid in the chain is always formylmethionine. And formylmethionine is simply the amino acid methionine, but with a formal group attached. And in case you don't remember what a formal group looks like, it looks like that. In eukaryotic cells, the first amino acid in all the polypeptide chains is simply methionine. And it's interesting to note that formylmethionine is actually acts as an alarm system in the human body. So if you had some bacterial cells in your body that were damaged in any way, there would be these formylmethionines floating around. And that tells your body that there are bacteria around and it's going to trigger an immune response.
Let's pause it and try to find the video with this guy. Let's duplicate this. Let's go to his channel. And let's type in translation your genome. Let's just watch a couple of these. This one's pretty good. Alright, let's do this, guys. I think that's it. Yeah, you can watch this one too. This one's 20 minutes long. Why is this 20 minutes long? Oh, this is nice. Oh, we'll watch this one too then. We'll watch these real quick before we move on. Well, not real quick, but let's take 10 minutes. Just gotta make sure it's in the brain. In the brain. Gotta get in the brain. Gotta get in the brain. Let's run it till 10 p.m. Yeah. Let's get it. Let's freaking get it, dude. Let's get it. Let's go after it. Huh? Okay. Let me mute the top. Oh, yeah, let me mute it. Put it. Hey, Lee, do you want to go see a movie with them? I can't go till 10. So if you want to go, you can go.
Which movie?
You're gonna see some Black Panther or something? Black something? I don't know.
Who?
Huh?
Your mom, Dad, and David?
Yeah. We can just go with David, I guess. Okay. Maybe it's at 7:30 or 8. Let me know because they want to know.
PM?
Yeah. 7:30 or 8, and it's the black guy, the center. Vampire.
No. Who's going?
They're all going.
Except you?
Yeah, I'm sorry. Because I messed up yesterday. I messed up. I don't know what happened. I think I left out like 3 or 4 hours or something. Really bad. I don't know how. It feels like I was like 3 hours behind. Today's gonna be a good day though.
Me too.
Huh?
Sure.
So you're gonna go? You can go. I'm just gonna be staying until 10 o'clock. 10 o'clock. Hello?
I'm saying I'm thinking because it's gonna be all Spanish.
David said speak Spanish. You speak with David. Or David can try to help you out. What the heck? Speaks the English. Just go, baby. Because I don't want you. Or you at work or what?
You don't want me
No,
here.
I mean you can't. I'm just gonna be unglued to the computer on my ass on C for the next 12 hours.
I don't want.
Okay. No? Or yeah?
Do I have to decide now? Just woke up. What's that?
Huh?
What's that?
Baby, how did it back? And then you want to know.
No.
Okay. You sure?
I don't want to know.
Babe, I'm gonna turn the lights on. Is that okay? Or no?
Yeah. Angelo, you say you're busy. I have to go. You're shaking.
Yeah, I gotta go. But I'm not busy to shake the ass. Shake the ass. Makes the ass clap.
And then on my phone.
Yeah.
That's not my phone.
Oh shit.
Oh shit.
Okay. Yeah, and then after that, today. Okay, I'm gonna unmute. Hey guys, we're back. It's kind of dark. Let me see if I can make it a little bit brighter. Hold on. Let's go.
Are those genes whose protein products either have a halting effect on the
Whoa,
regulation
whoa, 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 whoa. Slow down, lady. We're here.
After a polypeptide chain is formed, it's going to be folded into its secondary and tertiary structure into a very specific 3D conformation or shape. And at this point, we can start calling it a protein. But this protein may not be ready to carry out its function just yet. There might be some additional protein modifications that need to be made to this protein before it can be functional. And those are called protein modifications. There are two different types of protein modifications. The first type is co-translational modification.
Oh, you know, I
And
remember.
that
You guys can't see the screen. What am I doing? You guys can't see the screen. You guys gotta see what I'm learning too. Boom. Get it. Okay.
means that these are modifications or changes that happen to the protein or actually to the polypeptide while it's being translated. So let's see, we have the ribosome right here and we have a polypeptide that's being formed. So these changes are going to happen while the polypeptide is being formed. And an example of a co-translational modification is acetylation.
Wait, what did you say?
And what happens after a polypeptide chain is formed, it's going to be folded into its secondary and tertiary structure into a very specific 3D conformation or shape. And at this point, we can start calling it a protein. But this protein may not be ready to carry out its function just yet. There might be some additional protein modifications that need to be made to this protein before it can be functional. And those are called protein modifications. There are two different types of protein modifications. The first type is co-translational modification. And that means that these are modifications or changes that happen to the protein, or actually to the polypeptide, while it's being translated. So let's see, we have the ribosome right here, and we have a polypeptide that's being formed. So these changes are going to happen while the polypeptide is being formed. And an example of a co-translational modification is acetylation. And what happens during acetylation is the first amino acid in the polypeptide, which is usually methionine, is going to be removed.
Oh no.
And in its place, we put in acetyl group. So let's just draw an acetyl group. And acetylation happens to 80 to 90% of eukaryotic proteins. But the significance of this modification is not known very well. We're actually trying to figure out what the purpose of this modification is. The other type of protein modification that happens is post-translational modification. And actually, most protein modifications fall into this category, and the examples we're going to discuss in this video are all post-translational modifications, and those modifications happen after translation. Many post-translational modifications happen in the endoplasmic reticulum and the Golgi apparatus, but not all of them. So let's go through some examples.
Yes, ma'am.
So the first post-translational modification I want to talk about is glycosylation. And glycosylation, well, you can look at the word, the prefix glyco tells us that it has something to do with a carbohydrate. And so glycosylation is the adding of a carbohydrate to a protein. And most of the proteins in this video are all in green. And glycosylation usually happens is the adding of a carbohydrate to a protein. And most of the proteins in this video are all in green. And glycosylation usually happens to proteins that end up being embedded in the cell membrane. So you can see here we have a cell membrane and this protein embedded in it, and then there are these carbohydrate groups attached. So here's a carbohydrate group attached, and here's another one. And glycosylation helps to identify different types of cells. And one very common example of where we use glycosylation is in the ABO blood groups. So let's take 4 different red blood cells, and let's just say that each one of these red blood cells belongs to a different person. And so red blood cells have these proteins embedded in their surface. And these proteins are going to have many times different carbohydrate groups attached to them. So let's say that this person right here has this particular carbohydrate group attached to it. That makes him blood type A. Let's say that this person has a different type of carbohydrate attached to the protein. Let's say it looks something like that. That makes them blood type B. Let's say that this person has actually both of those carbohydrates attached to his red blood cells. That would make him blood type AB. And let's say that this last person does not have any carbohydrates of this category attach to the proteins on his red blood cells, and so that makes him blood type O. So here's a very common example of how glycosylation is used in the identification of different types of cells. Let's go on to a different type of post-translational modification that's pretty similar, and that is lipidation. That makes him blood type. So glycosylation in the different of these proteins. Glycosylation. Many post-translational modifications happen in the endo. So the first post-translational modification I want to talk about is glycosylation. A person attached to the protein membrane attaches very well to the this is on his red blood cells, and so that makes him blood type O. So here's a very common example of how glycosylation is used in the identification of different types of cells. Let's go on to a different type of post-translational modification that's pretty similar, and that is lipidation.
Lipidation.
And lipidation is when we add a lipid to a protein, also a protein that's going to be attached to the cell membrane. And this lipid we're looking at is actually an example of a GPI anchor. And GPI anchors are lipids that help to attach or tether proteins to the cell membrane. And just to give you an idea of maybe why this would be necessary, so to quickly review the structure of the cell membrane, we have these hydrophilic heads. That means that they are polar.
Polar, yes, ma'am.
And then we have inside these hydrophobic tails. And that means that they are nonpolar. And so the protein has both polar and nonpolar parts on it, and maybe it just doesn't attach well to the hydrophilic portion of the cell membrane. And so this GPI anchor, a lipid, kind of plunges into the lipid or hydrophobic part of the cell membrane. And we know that substances that are similar, like substances that are both hydrophobic, attach very well to each other. So this lipid, which is hydrophobic, attaches very well to the inner part of the cell membrane that is also hydrophobic. And so that's how it helps to attach the protein to the cell membrane. And both glycosylation and lipidation usually do occur in the endoplasmic reticulum or in the Golgi apparatus. Let's move on to some protein modifications that have more to do with the activity or the function of the enzyme and less with the structure. So one very, very common protein modification I want to discuss is phosphorylation.
Man, she's going through all of them, huh?
And phosphorylation is basically the adding of a phos, or the function of the enzyme and less with the structure. So one very, very common protein modification I want to discuss is phosphorylation. And phosphorylation is basically the adding of a phosphate group to a protein or to an enzyme. And then phosphorylation comes along with dephosphorylation is when you remove a phosphate group from the enzyme or the protein. I'm just going to make a little bit of room over here. And so what you're looking at is this schematic diagram of the sodium-potassium pump that's found in basically every animal cell. So the sodium-potassium pump. And another name for the sodium-potassium pump, which is the enzyme that you're looking at, is the NAKATPase.
NAK.
And so this enzyme or protein, the sodium-potassium pump, is responsible for maintaining the proper osmolarity of sodium ions and potassium ions in and out of the cell. And so let's see how phosphorylation regulates this protein, the cell, maintaining the proper osmolarity of sodium ions and potassium ions in and out of the cell. And so let's see how phosphorylation regulates this protein. So, and again, the proteins that you're looking at, they really all represent one protein. It's just we're going through the motions of the changes that happen to this one protein. But it's not like we have it's not like we're looking at 6 different proteins in the membrane.
Right, the whole thing.
So here's our first step. And when you look at this enzyme, you can see that there are 3 receptor sites for a particular ion. This is the sodium ion, represented by these dark blue circles. And then there are two receptor sites that look kind of more squarish, and those are receptor sites for the potassium ions, and those are represented by these light blue squares. So those are the potassium ions. And so back to the first step in what's happening here, the sodium ions will attach to the receptor sites on the enzyme. And just to clarify, this is the intracellular space. So this is basically the cytoplasm. And so these sodium ions are coming from the cytoplasm, and then out here is the extracellular space, the outside of the cell. Anyway, back to our first step. So the sodium ions attach to the receptor site, and when the receptor sites are full, it's going to cause something to happen. It's going to cause an ATP molecule, adenosine triphosphate, to break down into ADP, adenosine diphosphate plus phosphate. And then this phosphate will attach itself to the protein, and that is phosphorylation. And so when this protein gets phosphorylated, when the phosphate group is attached to it, it causes there to be some sort of change in the conformation of the protein. And that change in conformation causes the protein to turn itself around by 180 degrees and face the outside.
Oh, wow. And this does not actually cause cause the protein to turn 180 degrees. Instead, the shape of the channel running through the protein changes. So the shape of the channel. Oh, so it stays the same orientation going inside. But now it just kind of says, okay, you want to go out, go out. You phosphatated me. Now I'm going to put you outside. Kind of.
The cell.
Okay.
That's what you're looking at right over here. So our protein is still phosphorylated.
It looks like here, this kind of closes, but it can open, right? So
do it in a different place, but just, keep in mind that there's still a phosphate group attached to that protein. And then the protein releases these sodium ions into the extracellular space. So the next step is number 4, and again, we are still phosphorylated. There's still a phosphate group attached to our protein. And so in the next step, the potassium ions on the outside of the cells will attach themselves to the protein on the receptor sites. And take note, there are 3 receptor sites for sodium, but only 2 for potassium. When the potassium receptor sites are full, it's going to cause a different change to happen. This phosphate group is going to be removed.
Wait.
There are 3 receptor sites for sodium, but only 2 for potassium. When the potassium receptor sites are full, it's going to cause a different change to happen. This phosphate group is going to be removed. And that is called dephosphorylation. So that is dephosphorylation. And so that phosphate group ends up in the inside of the cell and it gets recycled in some other way. And when this protein is dephosphorylated, when that protein is removed, it's going to cause a different change in the conformation of the protein. And that change in conformation causes the protein now to turn around again by 180 degrees, and it faces the inside of the cell. So that's our 5th step. You can see the protein is facing the inside of the cell. And in the last step, the potassium ions are released into the inside part of the cell, and then we're back to our first step. And so you can see that the phosphorylation and dephosphorylation basically regulates the activity of this protein. And the end result that we're trying to get to is that we want there to be on the outside of the cell a rather high concentration of sodium, and a rather low concentration of potassium. And on the inside of the cell, we want there to be a rather low concentration of sodium and a relatively high concentration of potassium. And the end result that we're trying to get to is that we want there to be on the outside of the cell a rather low concentration of sodium, sorry, a rather high concentration of sodium, and a rather low concentration of potassium. And on the inside of this cell, we want there to be a rather low concentration of sodium and a relatively high concentration of potassium. And that's accomplished by the fact that for every 3 sodiums that are pumped out, 2 potassiums are pumped out. And a rather low concentration of potassium. And on the inside of the cell, we want there to be a rather low concentration of sodium and a relatively high concentration of potassium. And that's accomplished by the fact that for every 3 sodiums that are pumped out, 2 potassiums are pumped in.
Very nice.
And so here's an example, a very common example of how phosphorylation regulates a protein. And this doesn't happen just in the sodium-potassium pump. It happens in many, many enzymes and proteins in our bodies and in cells. Let's move on to 2 other protein modifications that also have to do with regulating an enzyme or something similar to that. So the next protein modification I want to talk about is methylation. And in particular, the methylation of certain proteins called histones. And those are these green circles that you're looking at. And histones are these proteins that around which DNA wraps itself. So they're found in the chromosomes and they help to package DNA in a very tight and organized manner. And sometimes histones are methylated. So let's put some methyl groups on our histones. And methylating and demethylating histones helps to turn certain genes on and off. And so here's another example of a protein modification helps to regulate activity, but in this case, we're regulating the activity of genes as opposed to proteins. And another protein modification I want to bring up is proteolysis. And by looking at this word, proteo means protein and lysis means to break something down or to cut something. And proteolysis is sometimes to take a protein and an example of how phosphorylation regulates a protein, and this doesn't happen just in the sodium-potassium pump. It happens in many, many enzymes and proteins in our bodies and in cells. Let's move on to 2 other protein modifications that also have to do with regulating an enzyme or something similar to that. So the next protein modification I want to talk about is methylation. And in particular, the methylation of certain proteins called histones. And those are these green circles that you're looking at. And histones are these proteins that around which DNA wraps itself. So they're found in the chromosomes and they help to package DNA in a very tight and organized manner. And sometimes histones are methylated. So let's put some methyl groups on our histones. And methylating and demethylating histones helps to turn certain genes on and off. And so here's another example of a protein modification helps to regulate activity, but in this case, we're regulating the activity of genes as opposed to proteins. And another protein modification I want to bring up is proteolysis. And by looking at this word, proteo means protein and lysis means to break something down or to cut something. And proteolysis is sometimes to take a protein and activate it, we need to cut it. And in fact, the insulin has to be cut twice before it's activated. So let's cut this protein twice. And you may have actually heard of the term zymogen.
Precursor to
And a zymogen is
enzyme.
an inactive form of an enzyme. And sometimes the way to activate a zymogen is by cutting it, and that's proteolysis. There is one more protein modification I want to discuss. And that is sometimes we add a protein ubiquitin, well, to another protein. So that protein I just added is ubiquitin. And this process is called ubiquitination. And what ubiquitination does is it basically marks this green protein for degradation, or for take a protein and activate it, we need to cut it. And in fact, the insulin has to be cut twice before it's activated. So let's cut this protein twice. And you may have actually heard of the term zymogen. And a zymogen is an
Hey, Dad, this is
inactive form of an
Yankee
enzyme. And
before I
sometimes the
actually
way to
finish
activate a
the
zymogen
lesson,
is by
because
cutting
then
it,
it's gonna be
and
too
that's
much to try to figure it
proteolysis.
out again.
There is one more protein modification I want to discuss.
Really?
And sometimes the way to activate a zymogen is by cutting it, and that's proteolysis.
Pause. Hydrolase. Protease, serine hydrolase, or protease. Hydrolase.
There is one more protein modification I wanted to do. So let's cut this protein twice.
Yeah.
And you may have actually heard of the term zymogen. And a zymogen is an inactive form of an enzyme. And sometimes the way to activate a zymogen is by cutting it, and that's proteolysis. There is one more protein modification I want to discuss. And that is sometimes we add a protein ubiquitin, well, to another protein. So that protein I just added is ubiquitin.
Another protein?
Protein.
Yeah.
So that protein I just added is ubiquitin. And this process is called ubiquitination. And what ubiquitination does is it basically marks this green protein for degradation, or for breakdown.
With a tag.
So within a short while of being ubiquitinated, this protein is going to be destroyed for
Degradation or breakdown.
So within a short while, this ubiquitinated, this protein is going to be destroyed, and the different parts are going to be recycled. Let's just quickly review the post-translational modifications that we discussed. So we talked about glycosylation and lipidation. Those are two protein modifications that generally happen to proteins that end up being embedded in the cell membrane. So glycosylation was the adding of a carbohydrate group, which helps to identify certain cells, and lipidation is when you add a lipid to a protein that generally helps to anchor a protein to the cell membrane. Then we talked about phosphorylation, methylation, the protein that glycosylation was the. Let's just quickly review the post-translational modifications that we discussed. So we talked about glycosylation and lipidation. And those are two protein modifications that generally happen to proteins that end up being embedded in the cell membrane. So glycosylation was the adding of a carbohydrate group, which helps to identify certain cells. And lipidation is when you add a lipid to a protein that generally helps to anchor a protein to the cell membrane. Then we talked about phosphorylation, methylation.
I was like, just get methylation.
And
phosphorylation. And what? Proteolysis? And ubiquitination.
And these all had to do with activating or deactivating an enzyme, or genes. So phosphorylation was we brought the example of the sodium potassium pump, which is basically regulated with phosphorylation and dephosphorylation. Then we spoke about methylation, which can basically helps to turn on or turn off certain genes. And proteolysis is the way in which many enzymes are activated. And the last post-translational modification we talked about was ubiquitination. And ubiquitination marks a protein for this is the way in which many enzymes are on or turn off certain genes. And proteolysis is the way in which many enzymes are activated. And the last post-translational modification we talked about was ubiquitination and ortho sodium potassium. So phosphorylation was we brought the example of the sodium potassium pump, which is basically regulated with phosphorylation and dephosphorylation. Then we spoke about methylation, which can basically helps to turn on or turn off certain. And the last post-translational modification we talked about was ubiquitination. And ubiquitination marks a protein for degradation, and then the various parts are recycled.
Great. Thank you so much. Thank you so much. Not yet. I don't know why I had this one like that, but let's watch these videos again, and again. They're not that.
The
They're
job
not that bad.
in protein.
In order for food to get the energy, it has to be broken down, taken up by the epithelial cells of your stomach in the stomach acid. First, it's going to break down the food molecules. Then those food molecules are going to be used for transcription and then translation. Now let's see what she says. Where was the girl? I think it was somewhere.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive?
In the nucleus.
Instructions to make proteins are contained in our DNA.
In our DNA.
DNA contains
Tightly wound nucleotides.
genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule.
The region that goes for an RNA molecule.
This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to to make a polypeptide in translation to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information then in translation, which occurs in
rtRNA binds to the mRNA and to the codon.
the cytoplasm
And these get transferred.
of the cell. The information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind. Is
Elongation
RNA
and what?
Elongation
Oh,
and termination. Fortune is controlled by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA
5'
strand.
to 3'.
As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
But it's pre-mRNA.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added.
Guanine, methylated guanine, a lot of adenines.
This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can exons to produce moves the intron segments and joins the adjacent exons to produce this complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore.
So
And
beautiful.
enter the
Oh my God, that's a beautiful animation.
Plasm to begin translation.
Wow.
How is the information in the mature messenger RNA strand translated into a protein? The nitro.
Oh, I gotta be, but this is beautiful. This is beautiful. Nobody thinks it's beautiful? I think it's beautiful. Okay, let's go back. Sorry about that, guys.
Nitrogenous bases are grouped into 3-letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon and binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down.
Oh, the complex slides down.
On one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum. Modified as it passes through the be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum. Modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
Nice. That's a good visual.
Proteins are
Hold on.
contained in our DNA. DNA
It's the
to
same
this
video.
site by the transcript is complete of messenger and modification
Promoter and terminator end.
RNA polymerides to the
Protein.
RNA molecule during each processing RNA found near the promoter for reaching access to this
Okay. Yeah.
begins.
Of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein. The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters
Yeah.
known as a codon. Each code letters known as of 3 letters in groups of 3 letters
Boom,
known
boom,
as a
boom. It's a
codon.
machine. It's an
Each
unstoppable
codon
machine.
on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA.
But where does the initial AUG methionine? Where does the initial AUG met go into the site of the large ribosomal unit? The A. I guess the E. A, P, or, I'm putting A or P, right? It's not going to go on E. It enters the P site, not the A site. Oh, it enters the P site right away. Boom, P site.
Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
quite literally turned into flesh and blood. Quite literally turned into flesh and blood.
Process in action. tRNA's enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. The basic unit nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA.
Wait, so it's 23 pairs. Oh, 23 pairs of chromosomes is 46 chromosomes. Hmm. 48 chromosomes.
DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes, which is to the start of the gene. This process for the ribosome reads the code in the messenger genome. A delivers the chain fork. Point for the when a gene is switched on, an enzyme called and acts as a template.
Oh, okay, hold on.
stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino codon of the trans molecule it binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes 1 intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay, hold on, guys. watched. And this one we already watched as well. Okay, let's keep this on here. Let's go ahead and watch. This was the next one. Let's go ahead and watch. Let's go ahead and watch these.
What I want to think about in this video is cell size, and in particular, how small cells can get, and then also what tends to be the limiting factors for how large a cell can get. And I have some pictures of cells here. This picture right over here, this picture of Pseudomonas bacteria. Each of these pill-shaped things, this is a bacterial cell. And just to get a sense of scale, the width of this pill is around 1 micrometer. So this is approximately 1 micrometer, which is the same thing as 1/1,000,000th of a meter, or you can think of it as 1,000th of a millimeter, whatever helps you conceptualize this better. And then the length here, this is about 5 micrometers. This is approximately 5 micrometers. Now over here, I have some pictures of cells that you would find in the human body. These are red blood cells. These have a diameter of about 7 micrometers. You see a similar scale for these white blood cells or some other things in here. Over here, we see a human sperm cell about to penetrate a human egg cell. And human egg cells are some of the largest. You might find it 5 micrometer, 1,000 micrometer. The width of this pill is around 1 micrometer. So this is approximately these pill-shaped things. This is a bacterial cell. And just to get a sense of scale, the width of this pill is around 1 micrometer. So this is approximately 1 micrometer, which is the same thing as 1/1,000,000th of a meter, or you can think of it as 1,000th of a millimeter, whatever helps you conceptualize this better. And then the length here, this is about 5 micrometers. This is approximately 5 micrometers. Now over here, I have some pictures of cells that you would find in the human body. These are red blood cells. These have a diameter of about 7 micrometers. You see a similar scale for these white blood cells or some other things in here. Over here, we see a human sperm cell about to penetrate a human egg cell. And human egg cells are some of the largest cells you find, especially if we're talking about spherical cells. And this cell here, this is going to have a diameter on the order of 100 micrometers. So the first question and it's kind of neat that all of these pictures are almost on the same scale, so you can almost compare them. But the first question we ask is, well, how small can a cell get? Well, if you think about it, a cell is a living thing. It's actually quite complex. It has to have information, it has DNA, it has to be able to replicate itself. It has all this metabolic machinery. Okay. So I just did some reading, and the smallest cells observed, and they think this might be the smallest cell period, although there might be future ones that are discovered that are even smaller, are actually on the order of about a few hundred nanometers. Remember, 1,000 nanometers would be the width of this pill. So a few hundred nanometers, maybe something like that would be maybe 300 nanometers. These were the smallest cells discovered so far. And they're bacterial cells. They were discovered at the University of California, Berkeley. And we think that this is pretty close to the lower bound, because you got to remember, we have to store all of this genetic information and all this cellular machinery. So that stuff's complex, and you can only get so small. But what about the upper bound of cells? Well, one of the things that it tends to be the limiting factor, and there's other things as well, but it's the ratio of volume to surface area. And why does the ratio of volume to surface area matter? Well, because the surface is what interfaces the cell with its surroundings. It has to take in nutrients and take out the waste. So each unit of surface area, it has to process the inputs and the outputs for a certain volume of cells, or for a certain volume of the cell. And as we'll see, as the cell grows, the volume and surface area don't grow together. The volume increases faster than the surface area does. So as you grow, each unit of surface area has to handle the processing with the environment for more and more volume. At some point, it just can't handle it. It can't take in nutrients and get rid of waste fast enough. And to make that a little bit more tangible, let's think about it mathematically. So the volume of a sphere, let's say this is a sphere here, so let me make it a little bit more three-dimensional. If it has a radius r, its volume is going to be 4/3 pi r cubed. Now its surface area is going to be 4 pi r squared. Now let's calculate the ratio of volume to surface area. That's what we really care about. The ratio of volume to surface area is surface area in yellow to surface area is equal to 4/3 pi r cubed over 4 pi r squared. Now, luckily, this simplifies quite nicely. 4 divided by 4 is 1, pi divided by pi is 1, r to the third divided by r squared is just going to be r. So this all simplified very nicely to r over 3. And if we wanted to care about units, it would be cubic units of volume, or it would be cubic units divided by square units, whichever unit we're looking at. So this is going to be r over 3. So let's use this to think about what happens as a cell gets much larger. So for simplicity, let's focus on this white blood cell here, and just to make the math easy, let's assume that it has a radius. Let's assume it has a radius of 3 micrometers. I'm going to do this in color. You can see 3 micrometers. So in that case, for this cell, its volume to surface area is going to be 3. We could just say 3 micrometers divided by 3, but I'll put 3. We could say 3 micrometers divided by 3, which of course is just going to be 1 micrometer. But having a unit of 1 micrometer for volume of surface area doesn't really make a lot of sense. An equivalent unit would say 1 cubic micrometer per square micrometer, because we're doing volume to surface area. And obviously, if you let the units cancel, you do the dimensional analysis, you'd be just left with this micrometer. But this helps us conceptualize a little bit more, because it says that each square micrometer needs to handle cubic micrometer. A unit of 1 micrometer for volume of surface area doesn't really make a lot of sense. An equivalent unit would say 1 cubic micrometer per square micrometer, because we're doing volume to surface area. And obviously, if you let the units cancel, you do the dimensional analysis, you'd be just left with this micrometer. But this helps us conceptualize a little bit more, because it says that each square micrometer needs to handle 1 cubic micrometer of cellular volume. So each square micrometer, so a square micrometer for this guy over here is going to be around that size. It's going to handle the processing on average for 1 cubic micrometer of volume. All right, that seems not reasonable, and that's a reasonable size for a cell. But what if we were to increase things by a factor of 1,000? Or increase the radius by a factor of 1,000. So, and I'm obviously not drawing this to scale, but let's say we find some new organism, or we theorize some organism that has that's cellular radius, instead of being 3 micrometers, so this was 3 micrometers, it's 3,000 millionths of a meter. And just to be clear, this isn't ginormous by our scales, this would be 3 millimeters. It would be visible by the human eye. The kind of threshold of what the human eye can see is about 1/10 of a millimeter, which is 100 micrometers. This is approximately, or this is 1/10 of a millimeter. So in the right conditions, you could just barely see a human egg cell. But this right over here, this would be still small by our scales, but let's just think about what happens to the volume to surface area. 3,000 micrometers divided by 3, 3,000 micrometers divided by 3, we'd be left with, this is 1,000 micrometers, or even better, we could write this as 1,000 cubic micrometers per square micrometer, So now each square micrometer, in this case, it has to handle a cubic micrometer of volume, but now it has to handle 1,000 cubic micrometers of volume. So it has to handle much more volume. And that's going to break down. It's not going to be able to exchange the gases, exchange the nutrients, exchange the waste fast enough for the cell to function. So this is a very important ratio, volume to surface area for cells. And actually ends up, well, I'll just talk about cells in general. It actually tends to be an interesting thing as a lot of things grow, volume to surface area or mass or mass. Well, there's a lot of other ratios that are interesting, but this is one of them. Now, the other factor that will play in is as also as the cell gets larger, the machinery has to just traverse more distances. You have to transport things over longer distances, which also can become cumbersome. But the volume to surface area is a really interesting one to think about, why we don't tend to see very, very, very large, especially spherical cells. And the reason why I emphasize spherical cells is because you do see cells that are longer than even this scale, like nerve cells. And they get by with that. They have other adaptations, but one of them is to just be really skinny and long. So this is one way that they can maximize their surface area. So like that, this is a nerve cell. Other ways that you'll see cells that maximize their surface area is that they have a lot of things that kind of stick out to maximize. So cells are clearly not all spherical. So they can have other things that maximize their surface area like that. So there's a bunch of adaptations, but in general, modeling them as a sphere isn't a crazy thing to do. And this is why we don't tend to see cells much larger than a human egg cell. When we want to categorize life as we know it at a very high level, we can categorize it as either eukaryotic, or as a eukaryote, or as a prokaryote, prokaryote. And the largest distinction between a eukaryote and a prokaryote are membrane-bound structures that eukaryotes have that prokaryotes don't have. And the most noticeable of which is a membrane-bound nucleus. So in a eukaryote, the genetic information is going to be inside a membrane-bound nucleus. So this right over here, this is the nucleus. This is the nucleus, and you have your genetic information inside it. You have your DNA. Here, for a prokaryotic cell, you're going to have your DNA, and it might be bundled up into a section of the cell. We would call that a nucleoid, but it's not membrane-bound. So let me write this down. No membrane. When we want to categorize eukaryote, the genetic information is going to be inside a membrane-bound nucleus. So this right over here, this is the nucleus. This is a nucleus, and you have your genetic information inside it. You have your DNA. Here, for a prokaryotic cell, you're going to have your DNA, and it might be bundled up into a section of the cell. We would call that a nucleoid, but it's not membrane-bound. So let me write this down. No membrane-bound nut or kernel. So let me make this clear. So this is referring to a nut, or a kernel, while prokaryote means before the nut or kernel. So we don't see, you just think of the nut or the kernel as the membrane-bound structure, especially the membrane-bound nucleus over here. But that's not all. Eukaryotes will also have other membrane-bound structures that you will not see in prokaryotes. For example, you will tend to see mitochondria. in a eukaryotic cell, both plant and animal cells, but you won't see it in a prokaryotic cell. And there's other types of membrane-bound structures. You can see things like Golgi apparatus. This over here is a micrograph of a eukaryotic cell, and you see the Golgi apparatus right over here, which helps package proteins. You see a micrograph of mitochondria. This is a micrograph of the nucleus. So this right over here is the nuclear membrane, and then you see all the genetic information. It's all spread out in chromatin form right over here. You see it's especially densely packed right over here. And we've also depicted this right over there. So this is all the DNA in chromatin form, but this part that looks extra dense or dark in this micrograph right over there, we call that the nucleolus. We call that the nucleolus. And we now know that this is where ribosomal RNA is being produced. And ribosomal RNA, that forms part of the structure of ribosomes, which are essential in the translation, or I should say the construction of proteins based on the information in mRNA. We go into a lot more depth of that in other videos. So these are ribosomes right over here, and they're made up of ribosomal RNA, and they're also made up of proteins. And so this nucleolus over there, that's where that's happening, the denser part of the nucleus. So the key distinction, eukaryotic cell, you have a membrane-bound nucleus. You have other membrane-bound structures like mitochondria. In fact, there's some theories that mitochondria first evolved as prokaryotic organisms that eventually lived in symbiosis inside of a larger eukaryotic cell. And the other distinction is that in eukaryotes, the DNA tends to be in multiple strands. So the DNA, if you were to kind of straighten it out, it would be in multiple strands. Well, in a prokaryote, the DNA tends to be circular. It can be all flipped around and whatever else, but at the end of the day, it would be circular DNA. So those are the three core distinctions: nuclear membrane, other membrane-bound organelles like mitochondria and Golgi and the Golgi apparatus, and then you also have multiple single strands of DNA versus circular DNA. Other things is that eukaryotes tend to be larger, while prokaryotes tend to be smaller. They tend to be simpler. So now that we know the key distinctions, what are examples of eukaryotes? Well, eukaryotes include most of what we interact with on a daily basis, or we think we're interacting with on a daily basis. This includes all multicellular organisms. So multicellular organisms. So I'm thinking animals, plants, fungi. It includes protists. This is paramecium right over here. This is eukaryotic. It's going to have a membrane-bound nucleus and other organelles. This right over here, these are onion root tip cells. So these are plant cells, and you can actually see it's been stained. You can actually see the membrane-bound nucleus here. This is actually a cool picture because you can see these cells in different stages of mitosis, which is interesting. Animal cells, the things that make you are eukaryotic. So what is prokaryotic? Well, bacteria is probably the most common example of that. Bacteria right over here. These are life. People, the current thought is that you have bacteria here, animals, and fungi, and cellular eukaryotes like protist.
Important to know. It's fine.
We are now going to talk about what is, when we've talked about macronucleic oxygen, that DNA information is stored in it. We're missing basic RNA. Totally got it enclosed in some type.
Cytosine, and guanine, which are pyrimidines. What? Mess it up. Oh wait, hold on. No. Oh my god. Adenine and guanine are purines. Adenine and guanine. But this is guanine and cytosine. So guanine is this one, which is 2, which is a purine, and then 1 is the pyrimidine, which I'm guessing is a cytosine. Okay, my bad. Well, it has A and U, so that's RNA.
We've already paid a lot of attention to the molecular structure of DNA. In fact, right in front of us, we have 2 strands of DNA forming a double helix, and we can look at the telltale signs that this is DNA. And in particular, we can look at the 5-carbon sugar on its backbone. We see, and let's actually number the carbons. This is 1', 2', 3', 4', 5'. We can see on the 2' carbon, we don't have an oxygen attached to it. We don't have a hydroxyl group attached to it. And because of that, we know that this is not ribose, this is deoxyribose. This right over here is deoxyribose, and these 2 are also deoxyribose. So that tells us that we have 2 strands of DNA, deoxyribonucleic acid.
Okay. Yeah. So watch this one through, and then we'll watch this one, and then we'll go back, finish this one, edit, cross it off, and then continue. I want to take the 10-minute break here for now, though. We're back. So let's go ahead and finish up these real quick, and then we'll add it to the deck.
From a deoxyribose being attached to 5', we can see on the 2' carbon we don't have an oxygen attached to it. We don't have a hydroxyl group attached to it. And because of that, we know that this is not ribose, this is deoxyribose. This right over here is deoxyribose, and these two are also deoxyribose. So that tells us that we have 2 strands of DNA, deoxyribonucleic acid. Let me write this down. This part of the chain, this is derived from a deoxyribose being attached to phosphate groups at a nitrogenous base. So deoxyribose. So what would we have to do if we wanted, instead of viewing this as a double, as 2 strands of DNA in a double helix formation, well, how would we have to rearrange, how would we have to edit the left-hand strand if instead we wanted to imagine that the left-hand strand is, say, messenger RNA being generated during transcription with a single strand of DNA here on the right? Well, to turn this into RNA, or to make it look like RNA, on the 2' carbon, well, we want to turn the deoxyribose into just ribose, so we would want to add a hydroxyl group right over here. So I add a hydroxyl group over there. Actually, do that. Do the hydrogens in white. So add 1 hydroxyl group there. And I want to do it on all the sugars on the left strand's backbone. If I want this to be a single strand of RNA, and RNA tends to be single-stranded. So oxygen and then a hydrogen. And so this hydroxyl, adding this hydroxyl group, instead of just having another hydrogen, just a hydrogen by itself over there, this tells us that this sugar is no longer deoxyribose. This is ribose. So now we have ribose. We now have ribose in our backbone, which is a telltale sign that, well, at least now we have the backbone of RNA, ribonucleic acid, versus DNA, deoxyribonucleic acid. Now, you might think we're done, but we're not quite done, because the nitrogenous bases on RNA are slightly different than the nitrogenous bases on DNA. On DNA, your nitrogenous bases are adenine, guanine, and adenine and guanine are the 2-ringed nitrogenous bases right over here. This is adenine, this is guanine, and you also have cytosine. Cytosine, I'm going to do these all in different colors. Cytosine and cytosine and thymine. I'm getting to the punchline too fast. And this right over here is cytosine, and this is thymine. And cytosine and thymine are single-ringed nitrogenous bases. We call them pyrimidines. Adenine and guanine, we call them purines. This is a little bit of review. In RNA, in pyrimidines, adenine and guanine, we call them purines. This is a little bit of review. In RNA, you still have adenine, you still have guanine, you still have cytosine, but instead of thymine, you have a very close relative of thymine, and that is uracil. So the way that this is drawn right now, this nitrogenous base, remember when we started this video, it was double-stranded DNA. This nitrogenous base right over here is thymine, and it forms hydrogen bonds with adenine right over here. If I want to turn into uracil, I just have to get rid of this methyl group right over here. So if I just do this, and if I were to replace it with a hydrogen that is just implicitly bonded there, well, now I'm dealing with uracil. So you see that uracil and thymine are very close molecules, or very similar nitrogenous bases, and that's why they can play a very similar role. And it's still the case. And so what uracil pairs with, it pairs still with adenine, the same thing that thymine pairs with, and everything else is, of course, still the same. Now, an interesting question is, why uracil? Why not thymine? Or you could say, why thymine? Why not uracil? And based on what I've read, it actually turns out that uracil is a little bit more error prone. It might be able to bond with other things. When you're coding, it's a little bit less stable than thymine. And so uracil makes the RNA molecule, or actually makes the machinery of information transfer, it makes it less stable. It's a less stable I guess, way to transfer information. And based on what I've read in evolutionary history, RNA molecules, most people believe, predate DNA molecules. And then so in the early stages, you had a lot of change, and so uracil molecules were just fine, and there was a lot of errors and whatever else. But then once you had, I guess, information needed to be a little bit more persistent and a little less error prone, well, then thymine helped stabilize. Thymine helped stabilize things. There's also the view of, well, why has uracil stuck around? Well, RNA molecules, they have all of these roles in cells. Messenger RNA molecules are taking information from the DNA and getting it transcribed or getting it translated at the ribosome, but they shouldn't hang out forever. That you actually want them to be somewhat unstable. So it's an interesting question to think about. Why do we have uracil instead of thymine, or why do we have thymine instead of uracil? But this is one of the telltale signs of that we are now dealing with an RNA molecule. So now what we have on the left-hand side, now all of this business, actually, let me do some different color. All of this business, this strand right over here, we can now, the way it's drawn, we can now consider this an RNA molecule. And if we assume that this is happening during transcription, when a DNA molecule, where a single strand of DNA would want to replicate its information, then this over here would be mRNA, messenger RNA. And so what's going on here? Well, let's think about it. This one, the way it's the RNA, the messenger RNA, the way it's oriented, we have phosphate group, then we go to 5' carbon, 4', 3', then phosphate group, then 5', 4', 3', then phosphate group. So this is oriented 5' on top, 3' on the bottom, while this DNA molecule is oriented the other way. This is a 5' carbon, this is a 3' carbon. So we have phosphate, 3', 5' phosphate. So we have 3' is on top and 5' is on the bottom. So if we wanted to think about what's happening, maybe using the symbols for the nitrogenous bases, we could say, all right, we have our mRNA molecule here, and this is its 5' end, and this is its 3' end. And then the top nitrogenous base right over here, this is uracil. This is uracil. And then the second one over here, this is cytosine. So this is cytosine. This is cytosine right over here. And this is being transcribed from a DNA molecule, from this DNA molecule on the right-hand side. So this is DNA. And this DNA has an anti-parallel orientation. It's parallel, but it's kind of flipped over. The sugars are pointed in a different direction. So this is this is the 3' end. This is the 5' end. And we see that the uracil is hydrogen bonded to adenine right over here. So adenine, and I'll draw dotted lines to show the hydrogen bonds. And that the cytosine is hydrogen bonded to guanine. So this right over here, that is guanine. Actually, I'll do the hydrogen bonds in white. So, they are, actually there's multiple hydrogen bonds going on here. But just to be clear, this is mRNA, and on the right, we have DNA. This could be happening during transcription. This could be happening during, I'm having trouble changing colors. This could be happening during transcription. Now, what are the types of RNAs out there? We've talked about this in other videos. Well, you have messenger RNA, which is an important role in taking information from DNA and getting it eventually translated with the help of tRNAs and ribosomes. And though I've just mentioned another type of RNA, and that's transfer RNA. So transfer RNA, tRNA. And in the video, the overview video on transcription and translation, we talk about how tRNA does this, but it brings amino acids, it has amino acids attached on one end, and it has anticodons on the other end that essentially pair with, when we think about DNA, we think about, okay, mRNA or RNA in that area for a thing called RNA. RNA came first, the RNA molecules, and the DNA eventually evolved from RNA, but RNA stuck around because it's still very useful.
What is a non-coding RNA? Well, a non-coding RNA, or an mcRNA, as it is abbreviated, is a functional RNA molecule that actually skips this last step and is not translated into a protein. In other words, they just go directly from transcription into an RNA molecule and then go off to perform any number of vital functions within the cell. And there are many examples of non-coding RNAs, including microRNAs, ribosomal RNA, transfer RNA, the list goes on and on. And as we go through ribosomal RNA, transfer RNA, the list goes on and on. And as we go through each of these different types and examples of non-coding RNAs, you'll start to see that there's sort of an emerging theme here. And that is that most of these non-coding RNAs participate in either transcription or translation in one capacity or another. So let's start off with microRNAs. MicroRNAs, or miRNAs, function in transcriptional and post-transcriptional regulation of gene expression. And they do this by base pairing with complementary sequences within mRNA or messenger RNA molecules. And this usually results in gene silencing through translational repression or target degradation. In essence, the mRNA to which these microRNAs bind are prevented from being translated, or they are sent on a pathway for degradation. Now, the next set of non-coding RNAs that we'll be talking about are all involved in translation. The first of which is ribosomal RNA. Now, ribosomes are the cellular machinery used to translate mRNA into proteins, and it is made up of one type of RNA molecule, ribosomal RNA. Transfer RNAs are an adapter molecule that links the codons in an RNA into proteins, and it is made up of one type of RNA molecule. The first of which is ribosomal RNA. Now, ribosomes are the cellular machinery used to translate mRNA into proteins, and it is made up of one type of RNA molecule, ribosomal RNA. Transfer RNAs are an adapter molecule that links the codons in an mRNA strand to the corresponding amino acids. And so this is another type of non-coding RNA that you'll see in translation. The third type is called SNO RNA, which stands for small nucleolar RNA. And it's a class of small RNA molecules that guide covalent modifications of ribosomal RNA, transfer RNA, and small nuclear RNAs, primarily through methylation, which is the addition of methyl groups, or pseudouridylation, which is the addition of an isomer of the nucleoside uridine. Now, another class of non-coding RNAs are the small nuclear RNAs, or snRNAs, not to be confused with the SNO RNAs, the small nucleolar RNAs that we just talked about. Now, small nuclear RNAs get their name from the fact that the average length of these RNA molecules is approximately 150 nucleotides. And their primary function is in the processing of pre-mRNA in the nucleus. They also aid in the regulation of transcription factors or a particular RNA polymerase polymerase 2, as well as maintaining telomeres, which are the regions of repetitive nucleotide sequences at the end of a chromatid, which protects the end of the chromosome from deterioration during chromosomal replication. Now, snRNA can be associated with a set of specific proteins and form complexes that are called small And there is a special SNRP complex called the spliceosome, made up of 5 small nuclear RNAs and over 150 proteins, that is responsible for splicing or removing the introns contained in messenger RNA, which is a major step in the post-transcriptional modification that takes place in the nucleus of eukaryotes. And the way that the spliceosome does this is that it binds to specific sequences in the pre-messenger RNA strand and performs 2 sequential trans-esterification reactions that splice out the intron and then ligate the 2 exons to form a mature mRNA. So now you know a little bit more of some examples of non-coding RNAs and some of the functions that they perform within the cell. So what makes a cell that's located inside of your nose responsible for smelling, say, a slice of pizza, look and act differently from a cell that lines your gut and is responsible for absorbing the nutrients from that pizza? They have the exact same DNA, so the differences can't be attributed to that fact alone. The answer actually lies in the expression of that DNA, which genes are actively transcribed and which ones aren't. And there are several ways in which gene regulation occurs at the level of transcription. And so we're going to be talking about the main ones here. Now, let's draw out a hypothetical gene here. And associated with this gene is a sequence upstream, so towards the 3' region of the antisense strand, also called the template strand. And this sequence is called the promoter. And there's another sequence in between the promoter and the gene called the operator. The operator is the sequence of DNA to which a transcription factor protein can bind, and the promoter is the sequence of DNA to which the RNA polymerase binds. They're called the template strand. And this sequence is called the promoter. And there's another sequence in between the promoter and the gene called the operator. The operator is the sequence of DNA to which a transcription factor protein The operator is the sequence of DNA to which a transcription factor protein can bind. And the promoter is the sequence of DNA to which the RNA polymerase binds to start transcription. Now, first off, in prokaryotes, we have what are called general transcription factors, which are a class of proteins that bind to specific sites on DNA to activate transcription. General transcription factors plus RNA polymerase and another protein complex called the mediator multiple protein complex constitute the basic transcriptional apparatus, which positions RNA polymerase right at the start of a protein coding sequence or a gene, and then releases the polymerase to transcribe the messenger RNA from that DNA template. Now, there's another type of DNA binding protein called activators, and these enhance the interaction between RNA polymerase and a particular promoter, encouraging the expression of the gene. And activators can do this by increasing the attraction of RNA polymerase for the promoter through interactions with subunits of the RNA polymerase, or indirectly by changing the structure of the DNA. An example of an activator is the catabolite activator protein, or CAP. And this protein activates transcription of the lac operon in E. coli. In the case of the lac operon in E. coli, cyclic adenosine monophosphate, or CAMP, is produced during glucose starvation. And so this CAMP actually binds to the catabolite activator protein, or CAP, which causes a conformational change that allows the CAP protein to bind to a DNA site located adjacent to the promoter. And then this activator, the cap, then makes a direct protein-to-protein interaction with RNA polymerase that recruits the RNA polymerase to the promoter. Now, enhancers are sites on the DNA that are bound to by activators in order to loop the DNA in a certain way that brings a specific promoter to the initiation complex. And as the name implies, this enhances transcription of the genes in a particular gene cluster. And while enhancers are usually what are called cis-acting, cis meaning the same or acting on the same chromosome, an enhancer doesn't necessarily need to be particularly close to the gene that it acts on. And sometimes it's not even located on the same chromosome. Enhancers don't act on the promoter region itself, but are actually bound by activator proteins. And these activator proteins can interact with that mediator complex I mentioned earlier, which recruits RNA polymerase and the general transcription factors, which then can lead to transcription of the genes. So here I've drawn a little schematic of what it might look like to have the enhancer actually change the structure of the DNA so that the DNA is now looping around. Here you still have your promoter sequence, the operator sequence, the gene sequence, and the enhancer sequence, and having the DNA looped in such a way so that you could then recruit RNA polymerase, the transcription factors, the mediator protein complex, and then you have transcription begin of this gene here. Now let's talk about repressors. Repressors are proteins that bind to the operator, impeding RNA polymerase's progress along the strand and thus impeding the expression of the gene. Now, if an inducer, which is a molecule that initiates gene expression, is present, then it can actually interact with the repressor protein in such a way that causes it to detach from the operator. And then this frees up RNA polymerase to then transcribe the gene further down on the DNA strand. One example of a repressor protein is the repressor protein associated, again, with the lac operon operator, which prevents the transcription of genes used in lactose metabolism unless lactose, which is the inducer molecule, is present as an alternative energy source. Now, silencers are regions of DNA that are bound by repressor proteins in order to silence gene expression. And this mechanism is very similar to that of the enhancer sequences that I just talked about. And similarly, silencers can be located several bases upstream or downstream from the actual promoter of the gene. And when a repressor protein binds to the silencer region of the DNA, RNA polymerase is prevented from binding to the promoter region. Now, a few notes about the differences between prokaryotes and eukaryotes when it comes to transcriptional regulation. In prokaryotes, the regulation of transcription is really needed for the cell to be able to quickly adapt to the ever-changing outer environment that it is sitting in. The presence, the quantity, the type of nutrients actually determines which genes are expressed. And in order to do that, genes must be regulated in some sort of fashion. So a combination of activators, repressors, and fairly enhancers, at least in the case of prokaryotes, determines whether a gene is transcribed. In eukaryotes, transcriptional regulation tends to involve a combination of interactions between several transcription factors, which allows for a more sophisticated response to multiple conditions in the environment. And another major difference between eukaryotes and prokaryotes is the fact that eukaryotes have a nuclear envelope, which prevents the simultaneous transcription and translation of a particular gene. And this adds an extra spatial and temporal control of gene expression.
Okay.
let's talk about post-transcriptional regulation. And basically, once DNA has been transcribed into mRNA. And I've drawn out this little schematic for you here, and it kind of just shows you how a DNA strand has a corresponding RNA strand and then the mRNA strand afterwards. And I'll sort of explain what all the different colors and words mean in just a little bit. So once DNA is transcribed by RNA polymerase into the corresponding RNA strand, this RNA strand needs to get what I call a tidy little haircut and then don some protective outerwear before it can leave the comfort of the nucleus for its big debut into the cytoplasm in the form of a fully processed messenger RNA or mRNA strand. Now, keep in mind that this form of regulation occurs in eukaryotes only, and this modification also helps to stabilize the mRNA to protect it from premature degradation before it gets translated into a protein. So as you can see here, DNA gets transcribed one-to-one, base for base, into RNA. And you can see here that there are sections of the RNA that ultimately make it into the finished mRNA, these short segments which are termed exons. And they are the sequences that code for the ultimate protein product. And then there are short non-coding segments of RNA that get cut or spliced out. And this would be the haircut that I alluded to earlier. And so these are called introns. And this is accomplished by a large molecular entity called the spliceosome. So the spliceosome binds on either side of an intron, loops the intron into a circle, and then cleaves it off, and then ligates the two cut ends of the exposing exons together, kind of cinches them together. And an easy way to remember which sequences are exons, which ones are introns, is that exons exit the nucleus, and introns stay in the nucleus. So exons kind of stands for exit. Now, even though the mRNA has gotten its nice little haircut, it's not quite ready to leave the nucleus just yet. It has to grab what is called a 5' cap and a 3' poly A tail. Now, what are those things that I just mentioned? So a 5' cap refers to changes at the 5' end of the mRNA. And remember that this is the phosphate end of the nucleotide bases in the mRNA. And some people like to remember this as F for 5' and for phosphate. So that's how you can kind of keep the two ends straight. And capping at the 5' end converts this end of the mRNA to a 3' end by a 5' to 5' linkage, which basically just protects the mRNA from exonucleases, which degrade foreign RNA. The cap also promotes ribosomal binding for translation and also helps the regulation of nuclear export of the mRNA. Now, the poly A tail, that goes on the other end, the 3' end of the mRNA, which has the terminal hydroxyl group. And so what do I mean when I say poly A tail? Well, poly A tail refers to polyadenylation, in which multiple adenosine monophosphates, or basically adenine bases, are added to act as a buffer for exonucleases in order to increase the half-life of mRNA. For translate, remember that this is of the nucleophosphate, which basically just protects phosphate. So that's a 5 binding when I say poly A tail. Well, poly A tail refers to polyadenylation, in which multiple adenosine monophosphates, or basically adenine bases, are added to act as a buffer for exonucleases in order to increase the half-life of mRNA and, again, protect it from degradation. And so the purpose of the poly A tail is really very similar to the 5' cap, which is basically to protect from degradation, help with promoting translation, and regulating nuclear export. The poly A tail also does one more thing. It kind of just helps with transcription termination for the RNA polymerase that's transcribing the messenger RNA. Polyadenylation is catalyzed by an enzyme called polyadenylate polymerase, which adds the adenosine monophosphates using adenosine triphosphate as the substrate. And the poly A tail is built until it's about 250 or so nucleotides long. So overall, the 5' cap and poly A tail help to stabilize the mRNA for translation. That's the key point to take home from here. So once the mRNA has donned its cap and tail and had its intron spliced out, it's now ready to exit the nucleus to be translated into a protein. Now, additionally, there's one more type of RNA regulation called RNA editing, which is a process that results in sequence variation in RNA molecule and is catalyzed by various enzymes. RNA editing is relatively rare, and these events may include insertion, deletion, and base substitution of nucleotides within the edited RNA molecule. Now, one of these enzymes is called adenosine deaminase acting on RNA, or ADAR enzymes, which convert specific adenosine residues to inosine in an mRNA molecule by hydrolytic deamination. Another type of editing is called cytidine deaminase acting on RNA, or CDAR, which involves deamination of cytosine to uridine by cytidine deaminase. RNA editing is currently being extensively studied in relation to infectious diseases because the editing process alters viral enzymes and their function. So kind of an exciting new emerging concept in post-transcriptional regulation.
Great. Thank you so much. open up the bag. You can open it. Yeah. Thank you. Just 3 more. Three more, and then what did I say we do? Oh, and then we do these, and then I add it. I think you might do this right now. the grand and the dog ran and a the dog ran and the dog ran and a the dog ran and a and dog ran and ate the dog ran and ate the dog ran and ate the hat. Oh yeah, 3 letters each, huh? 3 letters each. Dog ran and ate the hat. I think we already read this, that's fine. I'm not gonna read it again. Alright, let's take a 10-minute before we finish these up. Oh man.
But there's one special case that doesn't quite fit neatly into the box of lytic or lysogenic, and that's what we're going to talk about. So you might have an understanding of viral replication, but there's one special case that doesn't quite fit neatly into the box of lytic or lysogenic, and that's what we're going to talk about. So that special case is called a retrovirus. So first, let's zoom in and take a look at some unique things about the retrovirus that make it different from other viruses. So first of all, it is an enveloped single-stranded RNA virus. And inside of this envelope, it also carries 3 special proteins. And right now, just be aware that there are 3 special proteins. I'll talk about them more when we get to each step where they're important. So as you know, enveloped viruses can enter in one of 2 ways, either through tricking receptors, receptor-mediated endocytosis, or through direct fusion. And it just so happens that in our example, and we're talking here about the retrovirus HIV, this retrovirus will enter the cell with direct fusion. So now that this nucleocapsid is inside the cell, it actually has to undergo a step called uncoding, where this purple capsid is dissolved. Oh, we forgot about the proteins, so let me redraw those in right now. So these are the proteins that were originally inside of that capsid. So everything inside of that coat is released. And this is where the first special step occurs. So we're going to say that this red protein is reverse transcriptase. So reverse transcriptase will hop on to the RNA and it reverse transcribes the RNA, which means that, so it reads from 5' to 3' end, and you will form complementary DNA, shown here in pink. And the reason it's called reverse transcription is usually you take DNA to make RNA, but in this case, you take RNA to make DNA. And because this is the complementary DNA strand, we call this cDNA, complementary. And then reverse transcriptase will work again on the same RNA to make another cDNA strand. Because it's the same exact code, it can recombine with the other cDNA strands to make a double-stranded DNA. And so now what happens is you have integrase coming along. And let's make integrase blue. So integrase comes along, clips off each of the 3' ends. So now these are slightly shorter on each end. And sorry, this is a bit hard to see because this strand's 3' end is over here, and the first one is actually clearly labeled as this is 3' end. And by clipping off those 3' sections, they form these sticky ends because unpaired DNA wants to be paired, and integrase has suddenly removed that part. And you might be wondering what happens to this RNA, and what happens is that it actually gets degraded by normal ribonuclease. So that's no longer there. And integrase does exactly what it says. It will follow this path and integrate this HIV DNA into the host's DNA. And one thing I just want to very quickly mention is that if I had drawn this to be super accurate, this would need to have a nucleus around it because the HIV retrovirus infects human eukaryotic cells, which have a nucleus. So it actually will travel through the nuclear membrane to get to the genome. And here, integrase helps viral DNA integrate with the host. Like it's saying, integrate. So just imagine this is all double-stranded, but just for simple drawing sake, this will just be one line. So this is viral DNA, and this is called the provirus state. So you can see that this is similar to the lysogenic cycle that we've talked about before. But unlike the regular lysogenic cycle, it's not dormant or latent. It actually does not have that repressor gene that typical lysogenic viruses have. So it is actively transcribed whenever the host's DNA is transcribed. So since the host cell thinks this is normal DNA, it will make RNA. And I just wanted to call this viral mRNA so you have an idea. The cell cannot tell that this mRNA shouldn't have happened. So this mRNA exits the nucleus, and these viral RNAs are now in the cytosol. Again, once this viral mRNA exits the nucleus and goes into the cytoplasm, it's just like any other RNA. And some of these will be translated into proteins, like the capsid proteins, and of course the 3 proteins that we began with, which are the reverse transcriptase, the integrase, and actually the last one we haven't yet mentioned is the protease. The green here is protease. And we're going to hold off a little bit on what protease does, but here it's formed. And you can see that you now have all the parts that can self-assemble into new viruses. So again, all of these viruses that are formed will have the RNA, the reverse transcriptase, the integrase, and the protease. So you'll notice that these are actually missing one thing, they're missing their envelope. And so they're called immature viruses. And unlike the typical lytic cycle, it doesn't just break open the membrane. In fact, it takes advantage of the membrane. And so these viruses will come along and they will bud off. So this one will enter here, and this one will enter here. Oops, and that's missing a border, I just realized. So there you go. And they will bud off. And that will be their envelope. And sorry, they're missing the protein thing. I'll just draw them in again. So again, these are still immature, right? And before they go on to infect other cells, they have to mature somehow. So what happens is that protease inside of here will cleave those other proteins to make sure that they're fully functional before the virus enters another cell and starts this cycle all over again. And so retroviruses replicating are a bit more complicated than traditional replication. So it's not just lysogenic or lytic, it actually has elements of both.
So let's start looking at a short sequence of DNA. And the letters I'm going to use, these are the shorthands for the various nucleotide bases that make up a sequence of DNA. So let's say that I have some thymine, cytosine, guanine, cytosine, thymine, adenine, thymine, let's throw another thymine in there. So that would be our sequence of DNA. And what would be the corresponding sequence of RNA that it would be transcribed into? If you remember this from previous videos, pause this video and try to figure that out. Sequence of DNA. And what would be the corresponding sequence of adenine, thymine, thymine. Let's throw another thymine in there. So that would be our sequence of DNA. And what would be the corresponding sequence of RNA that it would be transcribed into. If
This
you
possibly?
remember this from previous videos, pause this video and try to figure that out.
I got UUGCGU8BBUU.
Key thing to appreciate is if we're talking about base pairs in DNA, adenine pairs with thymine, cytosine pairs with guanine. But if we're talking about pairing into RNA, well then instead of thymine in the RNA, you would have uracil. So the RNA here is, well, the thymine in the DNA would correspond to an adenine in the RNA, adenine, guanine, cytosine, guanine, the uracil, adenine, adenine, adenine. So this process that we just did, this is transcription.
No, never mind.
Transcription. Transcription from DNA to RNA. Now, the next step, if we're talking about the whole process of how does this information actually have an effect on the body, is we're going to go from the RNA and translate that into a protein.
So I think this was the DNA. This was the DNA. This is the new strand. What I made here was a template. This is a template. Opposite. And then just replace the keys with a U.
Protein. And the way we do that, we've seen this in previous videos, is every 3 of these bases, that's a codon, and it codes for a particular amino acid. Now, to figure out what amino acid it codes for, we look at an amino acid translation table. And there's different types that you might see. This is the most typical type. So the first base is A, second base is A, third base is G. First base A, second base A, we're in this cell, third base is G. And so that will code for the amino acid lysine. So we could write LYS, short for lysine here. And we could have also gotten that from a different type of translation table. For example, you might see a circular one that looks like that. But we would have gotten the same result. AAG, start at the center, AAG codes for lysine. Then the next codon, and if you're getting as excited about this as I am, I encourage you to pause this video and try to keep translating this. Then this actually for the where some of these bases, maybe something gets inserted, maybe something gets deleted, maybe something gets swapped out. And so let's start with what's known as a point mutation. So let's say this C gets swapped out for an A. Well, if that happened, then on the RNA strand, all of a sudden, this would be a uracil. And if that is a uracil, this AAG would still be there, coding for lysine. But this second codon is now different. What would it now code for? Well, CUA, CUA. It'll now code for leucine instead of arginine. Leucine, LEU. This is fairly typical for a substitution mutation. It might change a particular amino acid, but sometimes it could be more significant. For example, if this G was swapped out for an A, then this C on the RNA would then be a U, and then what would happen? Well, this first codon would still code for lysine, but the second one would be UGA. UGA. Now, all of a sudden, it codes for the stop codon. And so the actual translation process would stop, which could be a very, very big deal if this DNA sequence, if the normal non-mutated polypeptide had to keep going on and on and on. Over here, it just happened to have a stop codon next. But you can imagine if they had just another 1,000 codons before the end, but all of a sudden you had a point mutation to stop early, that would significantly affect the protein that is coding for it. Now, another type of mutation that typically has a fairly significant effect is a frame shift mutation. And that's where something gets inserted or deleted and shifts everything. So for example, instead of the A being swapped in for the G, what if the A got inserted here? So then our sequence would look like this. T, T, C, and then we have A, and then you have G, C, T, G, C, T, A, T, T, T. So what just happened here, this was our original sequence, but the A got inserted here. It didn't replace the G, and so everything got shifted to the right. Now what are we coding for? Well, when we transcribe to RNA, this will be A, G, U, C, G, A, U, A. And now this first codon still codes for lysine. You've seen that multiple times. But what about this second codon? The second codon over here, UCG. UCG, that's serine. We got a different amino acid. And what's interesting is it's not just that one amino acid is changing. We're going to see that keeps happening. So now we have AUA. AUA. Here we have isoleucine. So isoleucine right over here, which is different than what we had before. We don't have a stop codon anymore. And we would keep going on and on. And so you can imagine a frame shift mutation where you either insert something or you take it out so that the whole frame gets shifted can have a dramatic impact on what it will transcribe and then translate for. Now, lucky for us, even though mutations are always going on, there are many proofreading mechanisms in biological systems to make them less frequent than they otherwise would be. And people are still understanding how these proofreading mechanisms fully happen. Another thing to appreciate is we often associate a mutation as being equal to a bad thing. And oftentimes it is a bad thing. What used to be a functional protein may no longer be a functional protein because the amino acids, the coding got stopped short, or there was a frame shift mutation, it's just coding for completely different things. So sometimes it could be very bad. And some diseases actually are caused by strange mutations like that show up. Oftentimes, the mutation might not be a big deal. Maybe something gets swapped out. Maybe only one amino acid changes and it doesn't really change the ability of the protein to do its job, in which case it doesn't matter. But every now and then, a mutation can actually be a good thing. In fact, we need the mutation in order to have variation in the population. And variation is what natural selection and evolution run off. If you don't have variation, then you're not going to have different things that get selected in different environments, and you're not going to have that gradual change over time. So big picture, hopefully you got a better appreciation for how transcription and then translation, write that down, appreciation of how that happens. We got appreciation of how to use these translation tables, but also how either a point mutation or frame shift mutation can eventually affect the protein that gets coded for.
Ah. Sixteen 14. Mm. Oh, it's closing up, friends. This is recording may go for different. Oh. absolutely one. Oh. Oh, I see. Oh. What? 60? Code used only one piece. I can't go back. Can I? Okay. Let's pause it for now. think so.
Okay, are you gonna eat?
Oh man, where'd I put the cord? Where are you? Oh, you're right here. So we can cross out 84 to 90. I need something this week. Maybe I'll just read it, but I'm not really gonna read it that much. That's fine. We can just leave it. Okay, but then it means I'm not gonna add it. On week 11. Week 11 is the other. Okay. Okay, and week 5. So I think maybe week 5. Maybe we can just add it. This kind of stuff I kind of did cold. here yet. No. Okay. It's fine. Oh, holy crap. Yeah, and then I'm gonna add some more today as well. I might fall a little bit behind, so maybe on Sunday or something I'll be hitting it. At least I added the cards. So now we'll do gene control. Yeah. That's good. Okay, so what we're gonna do here. Alright, hold on guys, before we start, let me just clean up real quick. Sorry, it's kind of messy. Alright, I'm gonna reset the clock here, to be honest. And then, I'll take a 10-minute before I hit this one again. All right, guys, we're back. Got a good break. Let's continue. We are on gene control. This is ground control to Major Tom.
Expression describes the very first genetic. So the Jacob-Manon model for gene expression describes the very first genetic regulatory mechanism to be understood clearly. And it was first described.
Oops. This one's basically full, eh? Yeah. These pages.
'By French biologists François Jacob and Jacques Monod.
François Jacob and Jacques Monod
Originated the idea of regulatory mechanism to be understood clearly. So the Jacob-Manon model for gene expression describes the very first genetic regulatory mechanism to be understood clearly. And it was first described by French biologist François Jacob. So the Jacob-Manon model for gene expression describes the very first genetic regulatory mechanism to be understood clearly. And it was first described by French biologists François Jacob and Jacques Monod, who originated the idea that the control of enzyme levels in cells occurs through the regulation of transcription. And they, along with another scientist, shared the 1965
Maybe can you close the door? Lock the door. Thanks, baby.
Nobel Prize in Medicine for their work on what is called the lac operon. Now, a little bit of background first. An operon is a unit of genomic DNA containing a cluster of genes that are under control of a single regulatory signal, otherwise known as a promoter. And these genes are co-transcribed into a single mRNA strand and either translated to containing a cluster of operon is a unit of genomic DNA containing a cluster of genes that are genomic DNA.
Unit of genomic DNA.
Containing a cluster of genes that are under control of a single regulatory signal, otherwise known as a promoter. And these genes are co-transcribed into a single mRNA strand and either translated together or undergo transsplicing to
Babe, can you give me a a lighter shirt, not cotton. This is too hot. Please. Thank you.
Create single mRNAs that are translated separately. So basically, genes in an operon are expressed either all together or not at all.
The shotgun method.
Now, the operon that I've drawn here happens to represent the lac operon, and the lac operon
High wasted. Like the old guys in the movies. Not too much.
operon is an example of an inducible set of genes, which operon is an example of an inducible set of genes.
Stop. Wait a minute.
Operon, and the lac operon is an example of an inducible set of genes, which are responsible for importing and breaking down the sugar molecule lactose, for importing and breaking down the sugar molecule lactose to use as a source of energy. So in the event that glucose, which is the ideal source of carbon and energy for a cell, if that's not available, then the cell has sort of a backup source of energy in the form of lactose. And you can see where the name lac operon comes from, because it is named for the inducer molecule for the operon. And what do I mean by inducer molecule? Well, it is the presence of lactose.
So in the presence of lactose,
that actually induces
so if lactose is present, then start the gene. Like start transcribing the gene. Well,
is the
promoter,
transcription of the genes
I guess which genes? Pink and purple? Or the whole thing?
in this lac operon, which I'll explain in just a little bit. So there are 3 coordinately regulated genes contained in the lac operon. You have the LAC-Z gene, which codes for an unitly regulated operon, the transcription of the genes in this lac operon, which I'll explain in just a little bit. So there are 3 coordinately regulated genes contained in the lac operon. You have the LAC-Z gene, which codes for an enzyme called
beta-galactosidase.
Galactosidase. which is a cytoplasmic enzyme that cleaves lactose
Cytoplasmic enzyme that will
enzyme
what?
called beta-galactosidase, which is a
All right, and you are a cytoplasmic enzyme that will cleave
cytoplasmic enzyme that cleaves lactose into glucose
lactose into
and galactose.
glucose and galactose. Okay.
The next gene is the LAC-Y gene,
All right, the LAC-Y gene.
which codes for lactose permease.
All right, code for the lactose permease.
Which is a
Okay, you are a cytoplasmic membrane protein. I can't hit my protein.
cytoplasmic membrane protein that transports
Transfers
lactose
lactose
into the cell.
to the cell. Okay,
And then finally,
what we have?
you have the LAC-A gene,
All right, we got the LAC-A gene.
which codes for
Codes for
digalactoside
galactoside transacetylase.
acetylase. Now, only the LAC-Z and the LAC-Y gene are actually needed for lactose catabolism.
Needed for lactose catabolism.
LAC-A, LAC-Z, and the LAC-Y gene are actually needed for lactose.
The breakdown of molecules to extract energy. Breakdown of molecules to extract energy. LAC-Y gets lactose into the cell. LAC-Z turns lactose into glucose plus galactose.
Catabolism.
And LAC-A.
LAC-A is known as lactose. The next gene, enzyme that are gene protein, which codes for thiagalactoside transacetylase. Now, only the LAC-Z and the LAC-Y gene are actually needed for lactose catabolism. LAC-A is not as important in terms of understanding how the LAC operon works. Now, besides these three structural genes, LAC, Z, Y, and A, there are two regulatory sequences contained in the LAC operon, and they are called the promoter, which promotes the gene in the LAC operon, and they are called the promoter, which is important in terms of understanding how the LAC operon works. Now, besides these three structural genes, LAC, Z, Y, and A, there are two regulatory sequences contained in the LAC operon, and they are called.
Wait, what? I want to make sure I get it right. Oh, the promoter is something different.
The promoter, which promotes the transcription of structural genes, if you will, and the promoter, which promotes.
Yeah. There's so much stuff here. It's crazy. What's that?
Why are you going to Vietnam?
Where?
You have a Vietnam trip. You've been to Vietnam?
No.
It's you, babe.
No, never. And I think it was table tennis before. Never happened. Oh, that's a good one.
To go around this. What the? What if you have work to do?
Promotes the transcription of structural genes, if you will, and then also the operator. And there.
He has it.
He has what?
He has the access.
We'll just resend it to him. He's probably lost, baby.
There are two operon, and they are called the promoter, which promotes the transcription of structural genes, if you will, and then also the operator. And there are two other regulatory sequences that lie just upstream of the LAC operon that are genes that encode for a repressor protein, and then you have the associated promoter for that repressor protein.
Mm.
So these are the structural genes here, and then here are the regulatory genes. The structural genes here, and then here. Here are the regulatory genes. Now, when glucose is readily available to the cell, the repressor protein is constitutively expressed, meaning that it is transcribed at baseline, and that is just the default. And this regulatory protein binds to the LAC operator, and this interferes with and represses the binding of RNA polymerase, which wants to bind here to the LAC promoter. And this prevents and represses the transcription of these genes for lactose metabolism. Now, when glucose is not readily available to the cell and an alternate source of energy is available in the form of lactose, then things start to change. First, lactose passively enters the cell at a pretty slow rate, and a metabolite of lactose called allolactose then binds to the repressor. And this alters the conformation of this repressor protein or its shape, and it causes it to sort of loosen up and fall off the operator. Now, remember that the RNA polymerase is bound to the promoter immediately upstream of the genes. With the repressor now gone, RNA polymerase is free to sort of picture it rolling down to transcribe all of the 3 genes, leading to higher levels of the encoded proteins. So then you have lactose permease, which allows more lactose to enter the cell. And then you have more beta-galactosidase, which can break down the lactose into galactose and glucose to be used for the cell's basic metabolic needs. Now, what happens if both glucose and lactose are present? Which one does the cell prefer? Well, in that case, the transport of glucose actually blocks the transport of the inducer of the LAC operon, the lactose, in a process that's called inducer exclusion. So actually the transport of glucose into the cell leads to the inducer of the LAC operon, the lactose, prefer. Well, in
Yeah. This protein will block the operon unless lactose is around.
In that case, the transport of glucose actually blocks the transport of the inducer of the lac operon, the lactose, in a process that's called inducer exclusion. So actually the transport of glucose into the cell leads to the formation of this protein intermediate that binds to the lactose permease and prevents it from bringing in any more lactose into the cell. So then you have decreased lactose, which leads to decreased repressor protein binding. So then the repressor protein then sort of latches back onto the operator there and prevents the transcription of the rest of the lac operon genes. Now, there are two key takeaway points from the lac operon model. The first is to realize that it is the interaction between the inducer and the repressor molecules that mediate gene expression. And the second idea is that the cell expends energy to make enzymes only when necessary. So there are inducible genes whose transcription is induced when a particular molecule is present. So there are inducible genes who
Oh, did I make a new chat? Oh, beautiful. Just name this one. I need to rename you. I want to rename you, buddy. Oh, here. Rename you to rename you to gene control. This is crown control to gene control. I'm looking through the glass. I think that's what they say. Whatever. Get back over there.
is trans.
What? Why am I on D location? Get out of here. I was just Huh? Hello? Where are you? Where are you? What? Where'd you go? What the? Did it add it or didn't add it? Oh, I think it just deleted itself or something. Oh, right here. Oh, it's right here. I think it's need to refresh it. Oh yeah, that's very much. Jesus, get rid of you. Ah, you know what? I need to go to slide drops. Yeah, that was so quick. Oh my god. My eyes. I can see the world. Where are my glasses at?
What?
Where are my sunglasses at? Did they fall? I'll be back. We're back. Oh no, that's not. Yeah. That's a repressor promoter, dude. Repressor protein. Oh, gotcha.
So hopefully by now you're familiar with the central dogma of molecular biology that tells us that DNA makes RNA in a process known as transcription, and RNA makes protein in a process known as translation. Let's take a look at two cells. Let's say that right over here we have an eye cell, and let's say that we have a skin cell. And so what makes the eye cell an eye cell, and what makes the skin cell a skin cell? So they both have the exact same DNA, because all the cells in our body or in any organism have the same DNA. And so what makes an eye cell and a skin cell is which genes are expressed in that cell. So in the eye cell, we have the expression of genes that make certain proteins that are unique to an eye cell. And in a skin cell, we have genes that are expressed and they make proteins that are unique to a skin cell. And so the question I want to focus on is, how does that happen? How do we regulate the expression of genes so that only those proteins that are necessary for the cell get expressed or are made? So let's just frame our question. So we have genes that are expressed, and what makes an eye cell and a skin cell is which genes are expressed in that cell. So in the eye cell, we have the expression of genes that make certain proteins that are unique to an eye cell. And in a skin cell, we have genes that are expressed and they make proteins that are unique to a skin cell. And so the question I want to focus on is, how does that happen? How do we regulate the expression of genes so that only those proteins that are necessary for the cell get expressed or are made? So let's just frame our question.
That's the question, right?
How is gene expression regulated? So let's look at the options that we have. Maybe gene expression is regulated at the protein level. What that means is that the DNA in each cell is all transcribed into RNA, and then all the RNA gets translated into proteins. So according to this model, you'd have in each cell all of the proteins coded for by the entire human genome, if we're talking about humans, but then only those proteins that are necessary for the cell get activated. So for example, in the eye cell, all the DNA gets transcribed into RNA and they all get transcribed into proteins, but only the specific eye proteins are activated and all the other proteins are inactivated. Maybe that's what happens. Maybe gene expression is regulated at the level of translation. That would mean that all the DNA in the cell is transcribed into RNA, but not all the RNA gets translated into the. Maybe gene expression is regulated at the level of translation. That would mean that all the DNA in the cell is transcribed into RNA, but not all the RNA gets translated into protein. Just the RNA that make proteins of that particular cell would get translated. And the third option we have is maybe gene expression is regulated at the level of transcription. And that would mean that not all the DNA gets transcribed into RNA. Only the DNA that codes for proteins for that specific cell is maybe gene expression is regulated at the level of transcription. And that would mean that not all the DNA gets transcribed into RNA. Only the DNA that codes for proteins for that specific cell would get transcribed into RNA. So for example, in the eye cell, you'd only have DNA transcribed if that DNA is a gene that codes for a particular eye protein. And the answer to our question is that usually gene expression is regulated at the level of transcription. And if we think about it, this should make sense because this is really the most effective way for a cell to make use of its resources. So let's take a look again at our, if gene expression was regulated at the protein level. Again, say we're talking about humans, ourselves. So that would mean that in each of our cells, all the DNA gets transcribed into RNA is regulated at the protein level. Again, say we're talking about humans, ourselves. So that would mean that in each of our cells, all the DNA gets transcribed into RNA, and then all of that gets made into protein. So we would have a tremendous amount of protein in our cells. And actually thinking about it, I don't even think there's room in one cell for all of the proteins coded for in the genome. But even if there was, that would be a huge waste of energy.
God, yeah.
It takes a lot of
Every
ATP to
single
put together
cell?
proteins. And so why would we want each cell to make a whole bunch of proteins that they'll never even use? So this is not very efficient. What about translation? If we regulated gene expression through translation, well, it's more efficient than the protein level, but it's still also not that efficient because that means we have a bunch of RNA that would never even get translated. So making RNA doesn't take as much energy as making protein, but still that would be a big waste of energy. And so it turns out that regulating gene expression at the transcription level is the most efficient because we're not making any RNA or any protein that we're not going to use. And we actually have a lot more to learn about how gene expression is regulated, but there's a particular model that we understand pretty well, thanks to the work of two French scientists by the name of, one of them was François Jacob, and the other one was Jacques Monod. And they discovered the mechanism of the lac operon. So we call it the Jacob-Monod lac operon. And lac stands for the word lactose. And the lac operon is found in the bacteria E. coli, so it's a prokaryotic cell. And the picture that you're looking at is a sketch of the lac operon. It's a section of DNA in E. coli. And let's just label the diagram so that we orient ourselves. So let's say that this is the coding strand, which means that this is the non-coding or the template strand. And if you recall, it's actually the non-coding or template strand that gets transcribed. And that's the reason that I color-coded the non-coding.
I thought this one was a template strand. This was the new strand. Or whatever. I'm over here messing up the colors.
Strand with the various genes. Each of these colors represent a gene, and we'll explain in a minute what they are. If I wanted to be more exact, maybe I really should have also color-coded the top because these two strands are complementary to each other. But I'm not going to fill it out. Just use your imagination and remember these are complementary. And I also just want to point out that I drew this transcript. Bubble because it's going to be easier for me to show you what's going on in that way, but the default is that these two strands are really stuck together, and you do not have this bubble.
I think they get maybe replication. Okay, morphology.
Bubble forming unless transcript color coded the non-coding strand with various genes. Each of these colors represent a gene, and we'll explain in a minute what they are. If I wanted to be more exact, maybe I really should have also color coded the top, because these two strands are complementary to each other. But I'm not going to fill it out. Just use your imagination and remember these are complementary. And I also just want to point out that I drew this transcription bubble because it's going to be easier for me to show you what's going on in that way. But the default is that these two strands are really stuck together, and you do not have this bubble forming unless transcription is happening. So just keep that in mind as we go along. Okay, so what is this lac operon? So before we talk about the details, the lac operon has a couple of genes that will make enzymes that help E. coli break down lactose. So let's take a look. So over here we have these 3 genes. They're called structural genes. It's not important for you to remember that. But these 3 genes, this here is the LAC-Z gene, this is the LAC-Y gene, and this is the LAC-A gene. And so if you recall, the sugar lactose gets broken down into glucose and galactose. So glucose and galactose are monosaccharides, and lactose is a disaccharide. And the LAC-Z, LAC-Y, and LAC-A genes are all each going to code for an enzyme that helps in the breakdown of lactose, or in the metabolism of lactose. So let's look at the LAC-Z gene. The LAC-Z gene codes for a protein, beta-galactosidase. And beta-galactosidase is the enzyme that actually breaks lactose down into glucose and galactose. The LAC-Y gene codes for the enzyme lactose permease. The LAC-Z gene codes for a protein, beta-galactosidase. And beta-galactosidase is the enzyme that actually breaks lactose down into glucose and galactose. The LAC-Y gene codes for the enzyme lactose permease. And lactose permease helps the cell bring lactose into the cell. And the LAC-A gene also codes for an enzyme that helps in lactose metabolism. We just won't focus on it because it's not as important as the LAC-Z and LAC-Y gene. So these genes are all needed for the metabolism of lactose. And let's just label this part over here right before these 3 genes. That's the start site. So if RNA polymerase was transcribing, that start site tells it here's where you should begin to transcribe. And then after the LAC-A gene, we have a stop region. So that tells RNA polymerase stop transcribing. And normally, E. coli uses glucose as its energy source. That's the default. However, if glucose is not available, or if it's suddenly inundated with lactose, it will want to break down lactose. But why should E. coli express LAC-Z, LAC-Y, and LAC-A in the absence of lactose? Right? We just explained before that would be a huge waste of energy. So the default situation is that these genes are not expressed. Let's see how that is. So over here we have a promoter site. And the promoter site is the place that the RNA polymerase kind of just sits. So let's label our RNA polymerase. That's the enzyme that puts together RNA. So it just sits there. And after the promoter site, we have the operator site. And on the operator site, there's a protein that also just sits there. And this is called a repressor. And you can see the repressor is kind of blocking the RNA polymerase. So normally the RNA polymerase would want to proceed in this direction and transcribe this entire area of the DNA, but the repressor doesn't allow it to do that. It just sits there and blocks transcription. And so again, this is the default situation that's in the cell, uses glucose as an energy source, and transcription of these genes is blocked. Well, what happens when there's suddenly a lot of lactose in the cell? So let's just draw some lactose molecules. I'm going to draw them as these little triangles, although that does not adequately represent what lactose looks like, but for now we'll just draw it like that.
Don't worry about it.
So we have a bunch of lactose floating in the cell. So what's going to happen is that a lactose molecule will attach itself to the repressor protein. This changes the conformation of the repressor protein somewhat, and that causes the repressor to come off the operator site. So let's just get rid of our repressor.
Get out of here. Get out of here.
And let's put it
You go
over
away.
here for now,
Ooga.
together with the lactose that was attached to it. Now, the path of RNA polymerase is open, so it moves in this direction and transcribes all these genes. We make beta-galactosidase, we make
Wow.
lactose permase, and we have all the enzymes that we need to metabolize lactose. Well, what happens when the level of lactose goes down and we broke
Metabolize.
down all of our lactose?
Catabolize.
So let's get rid of some of our lactoses. Well, now, while they're all taken care of,
Hold on, ladies,
this
go back.
is lock it for now, together with the lactose that was attached to it. Now, the path of RNA polymerase is open, so it moves in this direction and transcribes all these genes. We make beta-galactosidase, we make lactose permase, and we have all the enzymes that we need to metabolize lactose. Well, what happens when the level of lactose goes down and we broke down all of our lactose? So let's get rid of some of our lactoses. Well, now, while they're all taken care of, including this lactose that was attached to the repressor, and when the lactose comes off of the repressor, it changes the conformation of the repressor and causes it to go back onto the operator site. So let's put him back where he was.
Get back in there.
Now you can see the RNA polymerase again is blocked. And so there is no more transcription happening right over here. So let's just connect this to what we spoke about in the beginning. We said that the regulation of gene expression happens at the level of transcription. We only transcribe those genes that we need. And this is exactly what's happening over here. In the absence of lactose, these genes are not expressed and we're saving the energy. But when we have lactose around, these genes are expressed and we have the enzymes necessary for the metabolism of lactose.
Very nice.
So the regulation of gene expression
Wow.
can be
She goes from like
modulated.
a Boston accent. They go sound regulation. Having your voice.
At virtually any step in the process, from the
I think it's
initiation of
different. It's
transcription
woman.
all the way to post-trans.
It's
So the
different.
regulation of gene expression can be modulated at virtually any step in the process,
Hold
from
on, lady. You're
the
talking too fast.
So the regulation of gene expression can be modulated at virtually any step in the process, from
Okay. What happens at the transcription level?
the initiation of transcription all the way to post-translational modification of a protein.
Well, that's true. That is true. Them are the facts.
And every step in between. And it's the ability to regulate all these different steps that helps the cell to have the versatility and the adaptability of an efficient ninja, so that it expends energy to express the appropriate proteins only when needed. Or you can think of the cell as a lazy couch potato that wants to expend the least amount of energy as possible.
There we go.
So starting at the beginning of gene expression, let's talk about gene regulation as it pertains to DNA and chromatin regulation. Let's talk about the structure of DNA. DNA is packed into chromosomes in the form of chromatin. So starting at the beginning of gene expression, let's talk about gene regulation as it pertains to DNA and chromatin regulation. Let's talk about the structure of DNA. DNA is packed into chromosomes in the form of chromatin, also known as supercoiled DNA. And so chromatin is made up of DNA, histone proteins, and non-histone proteins. And there are repeating units in chromatin called nucleosomes, which are made up of DNA known as packed into chromosomes in the form of chromatin, also known as supercoiled DNA. And so chromatin is made up of DNA, histone proteins, and non-histone proteins. And there are repeating units in chromatin called nucleosomes, which are made up of 146 base pairs of double helical DNA that is wrapped around a core of 8 histones. And there are 4 different types of histones within this structure of 8 that you should be aware of. And they're named H2A, H2B, H3, and H4. That's just the nomenclature they've been given. Now, acetylation occurs at the amino terminal tails of these histone proteins by an enzyme called histone acetyltransferase, which I'll just abbreviate as HAT. And this is a reversible modification. So the acetylation of histones is sort of kept in balance by another enzyme that removes these acetyl groups, which is called histone deacetylase, or HDAC-HDAC. The acetylation of histones leads to uncoiling of this chromatin structure, and this allows it to be accessed by transcriptional machinery for the expression of genes. On the flip side of this, histone deacetylation leads to a condensed or closed structure of the chromatin and less transcription of those genes. When these modifications that regulate gene expression are inheritable, they are referred to as epigenetic regulation. So when it comes to gene expression in DNA, you can basically think of DNA as coming in 2 flavors: densely packed and transcriptionally inactive DNA, which is called heterochromatin, and then less dense transcriptionally active DNA, which is euchromatin. And I like to think of heterochromatin as being densely packed and hibernating, like heterochromatin and hibernating both begin with H, kind of like a bunch of densely packed bears that are closed off in their cave for the winter. Whereas euchromatin is waiting there with open arms, welcoming the transcriptional machinery to transcribe away. Now, often you will see that histone deacetylation is combined with another type of DNA regulatory mechanism, and that is DNA methylation. And this occurs in a process called gene silencing. And this is a more permanent method of sort of down-regulating the transcription of genes. And DNA methylation involves the addition of a methyl group, which is a carbon with 3 hydrogens, to the cytosine DNA nucleotides by an enzyme appropriately called methyltransferase. And this typically occurs in cytosine-rich sequences called CPG islands. Don't forget that cytosine pairs with G. Guanine, so that's why there are CG islands that you'll find. DNA methylation stably alters the expression of genes, and so it occurs as cells divide and differentiate from embryonic stem cells into specific tissues. And so this is essential for normal development and is associated with other processes such as genomic imprinting and X chromosome inactivation, topics for another discussion. And abnormal DNA methylation has been implicated in carcinogenesis, or the development of cancer. So you can see how the normal functioning of DNA methylation is a critical regulatory mechanism for normal development and is associated with other processes such as genomic imprinting, transferase. And this typically occurs in cytosine-rich sequences called CPG islands. Don't forget that. That cytosine pairs with G guanine, so that's why there are CG islands that you'll find. DNA methylation stably alters the expression of genes, and so it occurs as cells divide and differentiate from embryonary to transcribe away. Now, often you will see that histone deacetylation is combined with another type of DNA regulatory mechanism, and that is DNA methylation. And this occurs in a process called gene silencing. And this is a more permanent method of sort of downregulating the transcription of genes. And DNA methylation involves the addition of a methyl group, which is a carbon with 3 hydrogens, to the cytosine DNA nucleotides by an enzyme appropriately called methyltransferase. And this typically occurs in cytosine-rich sequences called CPG islands. Don't forget that cytosine pairs with G guanine, so that's why there are CG islands that you'll find. DNA methylation stably alters the expression of genes, and so it occurs as cells divide and differentiate from embryonic stem cells into specific tissues. And so this is essential for normal development and is associated with other processes such as genomic imprinting and X chromosome inactivation, topics for another discussion. And abnormal DNA methylation has been implicated in carcinogenesis, or the development of cancer. So you can see how the normal functioning of DNA methylation is a critical regulatory mechanism for our cells. Now, DNA methylation may affect the transcription of genes in 2 ways. First, the methylation of DNA itself may physically impede the binding of transcriptional proteins to the gene. And second, and likely more important, methylated DNA may be bound by proteins known as methyl CpG binding domain proteins, or MBDs for short. Now, MBD proteins can then recruit additional proteins to the locus or particular location in a chromosome, certain genes, such as histone deacetylases and other chromatin remodeling proteins, and this modifies the histones, forming condensed inactive heterochromatin that is basically transcriptionally silent. So it is basically transcriptional.
All right, 10-minute. Quick 10-minute. Hey guys, we're back. Bit of a long break, I know, but it is what it is. I had to do some things, unfortunately. Let's go back. I was here. I put this in. Let's read this, and then let's continue. And then we got 3 more after this. 3 more. Tomorrow review early in the morning, and yeah, we'll see. Let's write down the card for what we did so that I could put it in Anki and find it pretty quickly after for tonight. And then let's see if we can wake up pretty early tomorrow just so I can finish more Anki because I'm definitely in the egg. Okay. DNA will wrap around histone H2A. I think it was H2B, H3, and H4. Let's go! That was forming nucleosomes, which packs into chromosomes. Okay, 2 forms of chromatin. And I think chromatin is just the one side of the chromosome? Is chromatin just one side of the chromosome? I'm gonna say no. Let's take a look at this. Chromatin is what's wrapped around the chromosome. Chromatin makes up the chromosome essentially with DNA, and then the histones. The histone core, which is 8 histones, right? Histone core is 8 histones. Yeah, it's an 8 protein complex. 2 copies of each of the 4 core histone proteins: H2A, H2B, H3, and H4. Okay, so the histones will wrap around the histone core, which is 8 histones, 2 of each, H2A, H2B, H3, and H4. They make nucleosomes, wrap them around a bunch of them really tight. You make chromatin, and then make an X, you get a chromosome, telomeres at the edges, centromere in the middle, and this found inside the nucleus, in the nucleolus. Chromatid is the one side. I think, is the chromatid. Yeah. 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 Alright, we gotta go way fast. Euchromatin is loosely packed and gene expression is active, transcribes the genes, and associated enzyme. We're gonna write this again. Two forms of chromatin. We have chromatin type, structure, gene expression, and associated enzyme. We have euchromatin, which structure is loosely packed, which has to be right for the DNA polymerase to be able to replicate, or also for the RNA complex to be able to get in there as well, right? Active genes transcribed. I guess this is in the transcription phase, so mostly just transcribed. We got hat, and then heterochromatin. We have tightly packed. How did the lady say she remembered it?
And this is a reversible, and on the flip side of, and less gene heterochromatin, and then less dense transcriptionally active DNA, which is euchromatin. And I like to think of heterochromatin as being densely packed and hibernating, like
Hibernating bears. Heterochromatin, hibernating bears. Silence. And that's HDAC. Hat adds acetyl groups to the histones. Done by histone acetyltransferase, and this loosens DNA wrapping. DNA is more accessible. Thank you. That is chromatin. I think we're writing down. I don't have enough time. We got it around the brain.
prochromatin, and hibernating both begin with H, kind of like a bunch of densely packed bears that are closed off in their cave for the winter. Whereas euchromatin is waiting there with open arms, welcoming the transcriptional machinery to transcribe away. Now, often you will see that histone deacetylation is combined with another type of DNA regulatory mechanism, and that is DNA methylation. And this occurs in a process called gene silencing. And this is a more permanent method of sort of downregulating the transcription of genes. And DNA methylation involves the addition of a methyl group, which is a carbon with 3 hydrogens, to the cytosine DNA nucleotides by an enzyme appropriately called methyltransferase. And this typically occurs in cytosine-rich sequences called CpG islands. Don't forget that cytosine pairs with G, guanine, so that's why there are CG islands that you'll find. DNA methylation stably alters the expression of genes, and so it occurs as cells divide and differentiate from embryonic stem cells into specific tissues. And so this is essential for normal development and is associated with other processes such as genomic.
We got it, lady. Thank you. Thank you. I think the name was Iptra. Already did this one? Whatever, run it again.
So what makes a cell that's located inside of your nose responsible for smelling, say, a slice of pizza, look and act differently from a cell that lines your gut and is responsible for absorbing the nutrients from that pizza?
We're running it.
They have the exact same DNA, so the differences can't be attributed to that fact alone. The answer actually lies in the expression of that DNA, which genes are actively transcribed and which ones aren't. And there are several ways in which gene regulation occurs at the level of transcription, and so we're going to be talking about the main ones here. Now, let's draw out a hypothetical gene here, and associated with this gene is a sequence upstream, so towards the 3' region of the antisense strand, also called the template strand. And this sequence is called the promoter. And there's another sequence in between the promoter and the gene called the operator. The operator is the sequence of DNA to which a transcription factor protein can bind, and the promoter is the sequence of DNA to which the RNA polymerase binds to start transcription. Now, first off, in prokaryotes, we have what are called general transcription factors, which are a class of proteins that bind to specific sites on DNA to activate transcription. General transcription factors plus RNA polymerase and another protein complex called the mediator multiple protein complex constitute
Watch this
the
video
basic
last. Go through the rest of the lesson and then come back.
which positions RNA polymerase right at the start of a protein coding sequence or a gene, and then releases the polymerase to.
We can come back to this one. So we'll come back to it. Next. I didn't read this article. So let's read it. Excuse me. 1, 2, 4, 1, 2, 3, 4. And can you do more sequences than that? Like 1, 2, 4, 1, 3, 1, 2, 1, 2, 3, 4, 1, 4, 3, 2. Right? Oh, okay. Mm. Going red. Okay, we got micro RNA gene. Transcription is a hairpin. That's RNA. cardiac hypertrophy. Ribosomal small unit 40S and ribosomal large unit 60S. Start for nutrients. Cortisol. This is zymogen activities. Zymogenic activity. Okay. Oh, babe, can I get the soda?
So much diabetes melody.
Can you stop?
Diabetes
Kirk.
melodies.
Can you stop?
So much sugar, that's why I can't sleep. Taking all the trash to me.
Okay, thank you. So good. Which one was this? I think the lady said. The lady said earlier. I wouldn't, yeah. Have the red notebook, please? Thank you. Aww. I think it was around here. Sometimes hard. I'm not ready right now. Oh, these are the ones you already went over. Excuse me. Proteasome. Okay. get in the trash. If you get ubiquitin, you get in the trash. You get ubiquitin, you get in the trash. That's where the protein goes, in the trash.
Give me that. Give me the trash.
Marked for ubiquination. Proteins marked for ubiquination. Ubiquitin. A bunch of ubiquitins. Post-transcription regulation? I just already played the video.
Adenosine triphosphate on its cap and tail and had its intron substitution of nucleotides within the edited, which involve infectious diseases because the editing process alters viral enzymes and their function. So kind of an exciting new emerging concept in post-transcriptional regulation.
Did I already watch all these? How did I already watch all these? I'm confused.
And then go off non-coding RNAs, you'll start to see that there's sort of an emerging theme here. RNAs or mRNA or messenger RNA molecules. The first of which is ribosomal RNA. Now, ribosomes are the cellular machinery. It is a non-coding RNA. Well, a non-coding RNA, or an mcRNA, as it is abbreviated, is a functional RNA molecule that actually skips this last step and is not translated into a protein. In other words, they just go directly from transcription into an RNA molecule and then go off to perform any number of vital functions within the cell. And there are many examples of non-coding RNAs, including microRNAs, ribosomal RNA, transfer RNA, the list goes on and on. And as we go through each of these different types and examples of non-coding RNAs, you'll start to see that there's sort of an emerging theme here. And that is that most of these non-coding RNAs participate in either transcription or translation in one capacity or another. So let's start off with microRNAs. MicroRNAs, or mRNAs, function in transcriptional and post-transcriptional regulation of gene expression. And they do this by base pairing with complementary sequences, which is sort of an emerging theme and is not translated into a protein. In other words, they just go directly from transcription into an RNA molecule and then go off to perform any number of vital functions within the cell. And there are many examples of non-coding RNAs, including microRNAs, ribosomal RNA, transfer RNA, the list goes on and on.
Okay.
And as we go through each of these different types and examples of non-coding RNAs, you'll start to see that there's sort of an emerging theme here. And that is that most of these non-coding RNAs participate in either transcription or translation in one capacity or another. So let's start off with microRNAs. MicroRNAs, or mRNAs, function in transcriptional and post-transcriptional regulation of gene expression. And they do this by base pairing with complementary sequences within mRNA or messenger RNA molecules, transcriptional regulation of gene expression. And they do this by base pairing with complementary sequences within mRNA or messenger RNA molecules. And this usually base pairing with complementary sequences within mRNA or messenger RNA molecules.
Okay.
And this usually results in gene silencing through translational repression or target degradation. In essence, the mRNA to which these microRNAs bind are prevented from being translated, or they are sent on a pathway for degradation. Now, the next set of non-coding RNAs that we'll be talking about are all involved in translation. The first of which is ribosomal RNA. Now, ribosomes are the cellular machinery used to translate mRNA into proteins, and it is made up of one type of RNA molecule, ribosomal RNA. Transfer RNAs are an adapter molecule that links the codons in an mRNA strand to the corresponding amino acids. And so this is another type of non-coding RNA that you'll see in translation. The third type is called SNO RNA, which stands for small nucleolar RNA. And it's a class of small RNA molecules that guide covalent modifications of ribosomal RNA, transfer RNA, and
Stop. Oh.
small nuclear RNA.
Okay, yeah.
RNAs, primarily through methylation, which is the addition of methyl groups, or pseudouridylation, which is the addition of an isomer of the nucleoside uridine. Now, another class of non-coding RNAs are the small nuclear RNAs, or snRNAs, not to be confused with the SNO RNAs, the small nucleolar RNAs that we just talked about. Now, small nuclear RNAs get their name from the fact that the average length of these RNA molecules is approximately 150 nucleotides. And their primary function is in the processing of pre-mRNA in the nucleus. They also aid in the regulation of transcription factors or a particular RNA polymerase 2, as well as maintaining telomeres, which are the regions of repetitive nucleotide sequences at the end of a chromatid that protects the end of the chromosome from deterioration during chromosomal replication. Now, snRNA can be associated with a set of specific proteins and form complexes that are called small nuclear ribonucleoproteins, or snRNPs, or sometimes people just call them SNRPs. And there's a special SNRP complex called the spliceosome, made up of 5 small nuclear RNAs.
Oh no, the lady.
Over 150 proteins that is responsible for splicing or removing the introns contained in messenger RNA, which is a major step in the post-transcriptional modification that takes place in the nucleus of eukaryotes. And the way that the spliceosome does this is that it binds to specific sequences in the pre-messenger RNA strand and performs 2 sequential trans-esterification reactions that splice out the intron and then ligate the 2 exons to form a mature mRNA.
Great.
So now you know a little bit more of some examples of non-coding RNAs and some of the functions that they perform within the cell.
Non-coding RNA, sancRNA. Thank you. The guy said finish it and then go back.
Oncogenes are genes that code for proteins that normally direct cell growth. They start out as Oncogenes are genes that code for proteins that normally direct cell growth. They start out as proto-oncogenes, and then something happens to convert that proto-oncogene into a full-blown oncogene. In essence, some sort of tumor-inducing agent, or it could also be totally spontaneous. Now, proto-oncogenes code for proteins that help to regulate cell growth and differentiation, which makes sense since the essence of a tumor, whether it is a cancerous or a benign one, is unregulated cell growth. In fact, the words oncogene and oncology, which is the study of cancer, share the same root word, onkos, which is Greek for mass or bulk. Now, the products of these genes are often involved in signal transduction and the execution of mitogenic signals. And you should recall that a mitogen is a chemical substance that encourages a cell to start cell division, basically something that triggers mitosis. So how does a proto-oncogene make that switch into an oncogene? Well, there are 3 main mechanisms: deletion or point mutation, gene amplification or increased mRNA stability, and chromosomal rearrangement. So let's start with the first one. Deletion or point mutation encoding sequence of the DNA in the gene itself or within a regulatory region, such as a promoter region, can lead to either a protein that is produced in the same normal amounts but is hyperactive for some reason, or maybe there is some sort of loss of regulation and the normal protein is just overexpressed. Gene amplification, or an increase in mRNA stability that prolongs the existence of the mRNA and thus its activity in the cell, can lead to a normal protein that is overexpressed. And then finally, chromosomal rearrangement involves translocation of a gene to a nearby regulatory sequence that then causes this normal protein, the gene product, to be overexpressed. Or you could possibly have fusion to an actively transcribed gene which overexpresses the fusion protein or leads to a hyperactive fusion protein.
Yeah,
And so
overexpression.
really you can
Overexpression,
start to see a
overexpression.
theme here emerging where the key idea is that you have either a normal protein that is just overexpressed, basically too much of the normal protein, or you can have normal expression, but the protein itself is just a hyperactive one. Now, there are tons of
ADHD.
examples of oncogenes, including the SRC oncogene, RAS, MYC, receptor tyrosine kinase, and also cytoplasmic tyrosine kinases. So let's talk about each one of those here. SRC was the first confirmed oncogene, which was discovered in 1970 and was termed
Tyrosine is a phenylalanine, but with the OH group at the end. It's polar hydrophilic, attacks phosphate groups.
Do you have this?
Yeah.
This one?
Mm-hmm.
SRC for sarcoma, which is a tumor of mesenchymal cells or connective tissue cells. It was actually an oncogene discovered in a chicken retrovirus. And so SRC codes for a non-receptor protein tyrosine kinase. So it
Tyrosine kinase, so it's going to
phosphorylates specific tyrosine residues in
Oh my god, I'm so good. That's what I was thinking.
other proteins. Another example of an oncogene is the RAS oncogene, which codes for a small GTPase, which hydrolyzes GTP into GDP and phosphate. This protein is activated by growth factors, so it phosphorylates specific tyrosine residues in other proteins. Another example of an oncogene is the RAS oncogene, which codes for a small GTPase, which hydrolyzes GTP into GDP and phosphate. This protein is activated by growth factor signaling and functions like a binary switch, sort of an on-off switch in growth signaling pathways. Examples of downstream effectors of RAS includes the protein MAPK, which is a type of kinase that in turn regulates genes that mediate cell proliferation. You can see examples of RAS oncogene mutations in thyroid tumors, certain leukemias, and cancers of the pancreas and colon. The MYC oncogene codes for a transcription factor that induces cell proliferation. And there's a very common translocation involving MYC between chromosomes 8, where the MYC oncogene is found, and 14, which leads to a certain type of lymphoma called Burkitt's lymphoma. The RTK oncogene stands for receptor tyrosine kinase, which is responsible for adding phosphate groups to other proteins to turn them on or off. These are similar to the SRC gene, which is a non-receptor tyrosine kinase, the difference being the location of the protein, either at the cell surface as a receptor or not as a receptor. So say you have a cell surface receptor that receives a signal from outside of the cell. Well, then that signal gets propagated or transmitted into the cell via these receptor tyrosine kinases that add phosphate groups to the target protein, specifically on tyrosine residues. And they can cause cancer by turning a receptor constitutively or permanently on in the absence of signals from outside the cell. Some well-known examples of receptor tyrosine kinases are vascular endothelial growth factor, or VEGF, epidermal growth factor, or EGFR, and platelet-derived growth factor, PDGF. And finally, we have the cytoplasmic tyrosine kinases, which mediate responses to the activation of receptors of cell proliferation, migration, differentiation, and survival. A well-known example is the BCR-ABL gene in chronic myelogenous leukemia, or CML, also known as the Philadelphia chromosome. It is a fusion of parts of DNA from chromosome 22 and chromosome 9. So basically, a part of chromosome 22, which contains the BCR gene, fuses with a fragment from chromosome 9, which contains the ABL gene. When these two chromosome fragments fuse, the genes also fuse, creating this new gene, the BCR-ABL gene. The fused gene codes for a protein that displays high protein tyrosine kinase activity, which is actually due to the ABL half of the protein. And the unregulated expression of this protein activates other proteins that are involved in cell cycle and cell division, which causes a cell to grow and divide uncontrollably, basically becoming cancerous. So as a result, the Philadelphia chromosome is associated with chronic myelogenous leukemia, a certain type of leukemia, as I mentioned before, as well as other forms of leukemias, which are cancers of white blood cells.
Thank you very much. Thank you kindly. I mean, I guess that's kind of true, though. Oncogenes are mutated forms of proto-oncogenes. Proto-oncogenes, if they do not turn into direct normal cell growth. I mean, that's, yeah, I think that's the gist of it.
The cell cycle, or they can also tumor suppressor genes are those genes whose protein products either have a halting effect on the regulation of the cell cycle,
Hold on,
or
lady.
they can also.
I don't know what you're saying. I need to see what you're saying.
Tumor suppressor genes are those genes whose protein products either.
Tumor what? Okay, look.
Tumor suppressor
genes
are those genes whose protein products either have a halting effect on the regulation of the cell cycle,
okay.
or they can also promote apoptosis, or sometimes both. So in other words, these proteins are like big stop signs that act as safety checks to help stop the mistakes in cell division that could lead to uncontrolled cell growth in cancer. Now, there are several sort of categories of tumor suppressor proteins. Those that recognize DNA damage and either repair it or initiate programmed cell death, apoptosis, if it can't be repaired, so DNA repair proteins. Then there are those proteins that act as repressors of genes that are essential for the continuation of the cell cycle. So if these genes are actively repressed and thus not expressed, the cell cycle does not continue on. So you have cell cycle repressors. With tumor suppressors, there is this concept called the two-hit hypothesis, in which both alleles, and remember that alleles are basically the copies for a certain gene. And you have two copies for any given gene, one on the chromosome that you got from your mom and one on the chromosome that you got from your dad. Now, in the two-hit hypothesis, both alleles must be mutated before the effect is manifested. Gene, one on the chromosome. With tumor suppressors, there is this concept called the two-hit hypothesis, in which both alleles, and remember that alleles are basically the copies for a certain gene. And you have two copies for any given gene, one on the chromosome that you got from your mom and one on the chromosome that you got from your dad. Now, in the two-hit hypothesis, both alleles must be mutated before the effect is manifested, because if only one of the alleles for the gene is damaged, then you have this sort of backup second copy that can still produce the protective protein. So you need two hits, one hit for each of the alleles that you have.
Gotta hit them both.
Another way you can think of this is that in mutated alleles for the gene is two-hit hypothesis, both alleles must be mutated before the effect is manifested. Because if only one of the alleles for the gene is damaged, then you have this sort of backup second copy that can still produce the protective protein. So you need two hits, one hit for each of the alleles that you have. Another way you can think of this is that in mutated oncogenes, these alleles are typically dominant. So a mutation in only one of the alleles yields the cancerous phenotype. But with a mutated tumor suppressor allele, these mutations are recessive because both alleles must be mutated in order to lead to the cancerous phenotype. The two-hit hypothesis was first proposed with cases of retinoblastoma, a rapidly developing cancer that originates from the immature cells of the retina, the light-detecting tissue of your eye. And I'll write this as PRB for retinoblastoma protein. Now, the retinoblastoma protein prevents the cell from replicating when its DNA is damaged. And it does this by preventing progression of the cell cycle from G1 into the S phase, or synthesis phase. So the retinoblastoma protein binds and inhibits transcription factors, which normally push the cell into the S phase. And this complex acts as a growth suppressor, and so the cell remains in the G1 phase. This complex also attracts a histone deacetylase protein to the chromatin, which reduces transcription of S phase promoting factors. And you can remember this by recalling that histone deacetylase leads to chromatin condensation.
HDAC, heterochromatin, hybrid bears gene is not active or sleepy, right? And it's more tightly bound and accessible by DNA polymerase, RNA polymerase as well, right? DNA polymerase and RNA polymerase as well, yeah.
Or transcriptionally inactive
We're talking about transcription here, so yeah, yes.
chromatin. So recalling that histone deacetylase leads to chromatin condensation or transcriptionally inactive chromatin. So this also further suppresses DNA synthesis.
Right, okay.
Another very well-known tumor suppressor protein is the P53 protein. Homozygous loss of this protein is found in up to 65% of colon cancers, 50% of lung cancers, and also in breast cancer. So this is clearly a very critical tumor suppressor protein. And so P53 activates DNA repair proteins when DNA has sustained damage.
Good job,
and
P53.
It can also arrest growth by holding the cell cycle hostage, if you will, at the G1 to S regulation point. And this gives DNA repair proteins some time to fix the damage and allow for continuation of the cell cycle. So specifically, P53 binds DNA and activates several genes, including ones that code for a protein called P21, which binds the cyclin CDK or cyclin-dependent kinase complex, which is actually the complex responsible for pushing the cell from the G1 to S phase in the cell cycle. P53 also functions in the initiation of apoptosis if the damage to DNA is irreparable. One significant exception to the two-hit rule for tumor suppressor genes is with certain mutations of the P53 gene product, which can then result in what is called a dominant negative, meaning that a mutated P53 protein can prevent the protein product of the normal allele from functioning. So don't forget to sort of keep that in the back of your mind when you're thinking about tumor suppressor genes.
Don't forget to keep everything in the back of your head. Regulation of transcription, this one.
This is the polymerase to transcribe the messenger RNA from that DNA template. Well, for smelling, say, a slice of pizza, look and act differently from a cell that lines your gut and is responsible for absorbing the nutrients from that pizza. They have the exact same DNA, so the differences can't be attributed to that fact alone. The answer actually lies in the expression of that DNA, which genes are actively transcribed and which ones aren't. And there are several ways in which gene regulation occurs at the level of transcription, and so we're going to be talking about the main ones here. Now, let's draw out a hypothetical gene here, and associated with this gene is a sequence upstream, so towards the 3' region of the antisense strand, also called the template strand. And this sequence is called the promoter. And there's another sequence in between the promoter and the gene called the operator. The operator is the sequence of DNA to which a transcription factor protein can bind, and the promoter is the sequence of DNA to which the RNA polymerase binds to start transcription. Now, first off, in prokaryotes, we have what are called general transcription factors, which are a class of proteins that bind to specific sites on DNA to activate transcription. General transcription factors plus RNA polymerase and another protein complex called the mediator multiple protein complex constitute the basic transcriptional apparatus, which positions RNA polymerase right at the start of a protein coding sequence or gene, and then releases the polymerase to transcribe the messenger RNA from that DNA template. Now, there's another type of DNA binding protein called activators, and these enhance the interaction between RNA polymerase and a particular promoter, encouraging the expression of the gene. And activators can do this by increasing the attraction of RNA polymerase for the promoter through interactions with subunits of the RNA polymerase, or indirectly by changing the structure of the DNA. An example of an activator is the catabolite activator protein, or CAP, and this protein activates transcription of the lac operon in E. coli. In the case of the lac operon in E. coli, cyclic adenosine monophosphate, or CAMP, is produced during glucose starvation. And so this CAMP actually binds to the catabolite activator protein, or CAP, which causes a conformational change that allows the CAP protein to bind to a DNA site located adjacent to the promoter. And then this activator, the CAP, then makes a direct protein-to-protein interaction with RNA polymerase that recruits the RNA polymerase to the promoter. Now, enhancers are sites on the DNA that are bound to by activators in order to loop the DNA in a certain way that brings a specific promoter to the initiation complex. And as the name implies, this enhances transcription of the genes in a particular gene cluster. And while enhancers are usually what are called cis-acting, cis meaning the same or acting on the same chromosome, an enhancer doesn't necessarily need to be particularly close to the gene that it acts on. And sometimes it's not even located on the same chromosome. Enhancers don't act on the promoter region itself, but are actually bound by activator proteins. And these activator proteins can interact with that mediator complex I mentioned earlier, which recruits RNA polymerase and the general transcription factors, which then can lead to transcription of the genes. So here I've drawn a little schematic of what it might look like to have the enhancer actually change the structure of the DNA so that the DNA is now looping around. Here you still have your promoter sequence, the operator sequence, the gene sequence, and the enhancer sequence, and having the DNA loop in such a way so that you then recruit RNA polymerase, the transcription factors, the mediator protein complex, and then you have transcription begin of this gene here. Now let's talk about repressors. Repressors are proteins that bind to the operator, impeding RNA polymerase's progress along the strand and thus impeding the expression of the gene. Now, if an inducer, which is a molecule that initiates gene expression, is present, then it can actually interact with the repressor protein in such a way that causes it to detach from the operator. And then this frees up RNA polymerase to then transcribe the gene further down on the DNA strand. One example of a repressor protein is the repressor protein associated again with the lac operon operator, which prevents the transcription of genes used in lactose metabolism unless lactose, which is the inducer molecule, is present as an alternative energy source. Now, silencers are regions of DNA that are bound by repressor proteins in order to silence gene expression. And this mechanism is very similar to that of the enhancer sequences that I just talked about. And similarly, silencers can be located several bases upstream or downstream from the actual promoter of the gene. And when a repressor protein binds to the silencer region of the DNA, RNA polymerase is prevented from binding to the promoter region. Now, a few notes about the differences between prokaryotes and eukaryotes when it comes to transcriptional regulation. In prokaryotes, regulation of transcription is really needed for the cell to be able to quickly adapt to the ever-changing outer environment that it is sitting in. The presence, the quantity, the type of nutrients actually determines which genes are expressed. And in order to do that, genes must be regulated in some sort of fashion. So a combination of activators, repressors, and rarely enhancers, at least in the case of prokaryotes, determines whether a gene is transcribed. In eukaryotes, transcriptional regulation tends to involve a combination of interactions between several transcription factors, which allows for a more sophisticated response to multiple conditions in the environment. And another major difference between eukaryotes and prokaryotes is the fact that eukaryotes have a nuclear envelope, which prevents the simultaneous transcription and translation of a particular gene, and this adds an extra spatial and temporal control of gene expression.
Said tips or thanks, so I said thanks. Oh, 11 questions. 5 minute break. Yeah, just a quick 5 minute, quick 5 minute.
Babe, can you see this?
Yeah.
So